So, DK sir, shall we start now?
Yeah, please start. Yes.
Dr. Ajay, please start. You are audible.
Yes, okay. Thank you, sir. Good morning, everyone. On behalf of Amity Institute of Organic Agriculture, Amity University, Uttar Pradesh, Noida, I, Dr. Ajay Tomar, Assistant Professor Second, I extend a very warm welcomes to all the distinguished experts, scientists, faculty members, researchers, research scholars, and participants joining us for the Day 3 session first of the 5-day faculty development program on climate resilient agriculture and allied technology for net zero and socially inclusive framework. I would like to express our sincere gratitude, our visionary leadership for their continuous support and encouragement. We are deeply thankful to our Honorable Founder, President Sir Dr. Ashok K. Chauhan Sir, and Honorable Vice Chancellor Dr. Atul Chauhan Sir, Honorable Vice Chancellor Ma'am Dr. Balvinder Supra, Director General Amity Food and Agriculture Foundation, and Chancellor Dr. Nutan Kosik Ma'am, and Director Amity Institute of Organic Agriculture, Dr. Doili Sainal Ma'am, whose guidance has made this program possible. As we are the third day of the FDP, we continue our exploration of innovative approaches, emerging technologies, and scientific advancements that are transforming agriculture into a more sustainable, resilient, and inclusive sector. Today's session features eminent experts from the fields of molecular plant biotechnology. So it gives me a great pleasure to introduce our distinguished speaker, Dr. Niti Sanan Misra, Group Leader at the International Center for Genetic Engineering and Biotechnology. Dr. Misra is an internationally recognized molecular biologist and plant scientist whose pioneering research in RNA interference, microRNA biology, and plant stress responses has significantly advanced our understanding of crop adaptation under environmental stress conditions. Her research focuses on developing climate resilient crops through the cutting-edge approaches involving molecular biology, genomics, bioinformatics, and biotechnology. Her contribution continue to strengthen global efforts towards sustainable agriculture and food security. We are honored to have such an immense scientist with us today. Ma'am, the platform is all yours.
So a very good morning to all. And to start with, at the onset, I'd like to thank the organizers for asking me to present in this FTP. And I'm mainly going to talk about the basics of microRNAs and give you a little flavor of the kind of work that we are doing without actually getting into details of the data. So can I share my screen, please, so that the slides are visible?
Dr. Karthik, please. Yeah.
Are they visible?
Madam,
yes, ma'am. Visible.
Slideshow, maybe.
Yeah. You are on. I'm on the slideshow. Is it?
Yes,
please. For you? Okay. So, to align with the theme of the meeting or the workshop, I thought we'll talk about microRNAs as and their role in driving climate resilience and how these can be used as tools in the plants. Because food, is the major question for all of us. And the biggest challenge for growing this food, that is agriculture, is actually population. And this population, while the population is increasing, we need to understand that we have less and less of land and less of water as it is gradually decreasing. But still, the hard work of the farmers and the great breeders we managed to produce enough to feed the growing population. And with the increase in land, the rice production is actually, forecasted to increase. But then came the challenge of climate change. And this has really upset the predictions, because the globally rising temperatures are not only bringing high temperature stresses to the plants, but they are also affecting because of the irrigation practices, they are leading to buildup of salt in the soils, they are creating situations of drought, causing sea levels to rise, which bring up another challenge. And all this, especially the increase of soil in the salts, is becoming detrimental for agriculture, and we perceive a decrease in production potential up to about more than 46 million hectare per year annually globally. So this is a real serious challenge that needs to be addressed. But this is not a new problem. Scientists have always been aware of it and have been working on salinity stress for a long time now. And we know what exactly soil salt does to the plant. It creates a ionic osmotic stress, which affects a variety of different parameters in the plants. I'm not going to list all of them. And plants have inherent mechanisms to sort these out. They have mechanisms, there's a very well laid out methods of perception, signaling, and alteration of gene expression, so that the plants are able to withstand the stress to a threshold beyond which they are unable to cope and they die. Having said this, since the response creates a multi-genic challenge, the plants also involve a lot of different pathways and genes. So overall, it is a complex trait, and an intricate regulation is involved. It was thought that with the sequencing of the rice genome, probably we'll get more clarity into what are the genes and how these are regulated, so on and so forth. And with this concept or idea in mind, the first rice genome sequencing initiative started around 1998, and now we have several hundreds of different rice genomes at the draft and the full genome level, which are available. And the indica rice genomes were available by 2002. Now, why I'm speaking about these timelines is because it is this period, 1998 to 2002, which was very crucial, because this was the time when another great discovery happened, and that was the discovery of the small RNAs, or the RNA silencing. Because till that time, what we understood was that DNA codes for RNA, and RNA makes the protein, which are the functional molecules or the enzymes in the cell, and are responsible for the various phenomena that biological phenomena that are occurring, whether they are plants or animals. But now was introduced a concept where RNA, a regulatory role was added to the RNA, and this was not of expression, but for switching off gene silencing. And this really changed the dogma, because now we didn't have to overexpress something. We had to understand the balance in expression of how the genes are regulated. So just a quick introduction to these small RNAs for those of you who are not aware. I'm sure a large population is aware about the regulatory small RNAs. They are a pool of 21 to 24 nucleotide RNAs, and their major role is in gene silencing. It is also known as RNA interference in the non-plant systems. And this normally occurs at the post-transcriptional level, where the small RNA will go and bind to the transcript and would either cleave it or make it or repress its translation by causing steric interference. But they are also known now to be involved in the transcriptional gene silencing by bringing about epigenetic modifications at the chromatin level. So the general concept is that wherever a quick response is required, say a quick response to a stress or to a hormonal cue or something, then the PTGS pathway is operational. And wherever stable information needs to be transported from the parent plant to the progenies, or for long-term silencing where you don't want a gene to be expressed, then in that case, the organism will go through the TGS pathway. So you both have situations of stable and, or rather, we should say PTGS and TGS silencing pathways. So, like I said, 1998, the response, though, is well understood in nematodes and other non-plant system. The first observation came from plants, and it was first noted when there were a group of scientists in the Jorgensen's group were trying to do a simple experiment. You all must have seen the petunia flowers, which normally flower during these, the change of season from winter to summers, and they are bright colored flowers, large flowers, and are very easy to maintain. So they were very popular. So the group was trying to enhance the intensity of the flower color. And the idea seemed very simple, that since CHS, that is the chalcone synthase, is involved in production of the anthocyanin pathway, and more the anthocyanins, the darker would be the color. So if you overexpress the gene, you would tend to have more of the pigment accumulating, and hence can produce dark color flowers. And when they actually went and did an overexpression, to their surprise, they found different kinds of phenotypes from variegated to absolutely colorless ones. And this actually laid down the formation of sense-mediated silencing. Till now, it was only antisense gene-mediated silencing. Whenever we had to switch off a gene, put in the antisense constructs, and some magic would happen, and the gene would get switched off. The accepted hypothesis was that the two mRNAs would bind to each other, forming a double-stranded region, which double-stranded molecule, which is not able to be translated, and hence the gene expression switched off. But this was the first proof of sense-mediated silencing, where you were trying to overexpress a particular gene in presence of an endogenous environment, but instead of getting overexpression, a reverse, that is silencing of that gene, happened. And this triggered triggered the discoveries. I'm not going to get into the detail of that. And ultimately, in 1998, in C. elegans, it was observed for the LIN4 and the LET7 genes are targeting, which used to target the LIN14 genes. These are all developmental phenotypes which are present in the C. elegans. What I would like to highlight here is this, that at the L1, that is these are the larval stages, the L1, L2, and 4, so on. At the L1 stage, there was no expression of the LIN4 RNA. It came at the L2 stage. But at the L1, both at the L1 and L2 stage, they saw the presence of the LIN14 RNA. But to their surprise, at the L1 stage, LIN14 protein was present, which somehow disappeared at the L2 stage. And later on, a series of elegant experiments were done by different groups, including that of they discovered that this no protein was formed because the LIN4 was not coding for a protein. It was making a small RNA, and this small RNA was going and interfering with the transcription of LIN14, and this triggered the discoveries, and eventually the regulatory small RNAs were identified. But what it changed drastically was our understanding of how gene expression happened. It was not only expression regulated by the transcription factors, but the effect we see at the protein level is actually a balance between expression and silencing. So the cells can control which protein will be formed at what time and how much of it can be formed to bring about the optimum expression. And this has really unfolded another layer and everybody is every aspect of research can now be reinvestigated to understand what is happening at the different levels. Over the last 25 years, our understanding of the non-coding RNAs and the phenomena of RNAi or RNA silencing per se has increased dramatically. A lot of protein players have been identified in the pathway. We could identify different kinds of the small RNA molecules, which could be categorized based on the way they are origin, that is on their biogenesis. And we have categories of even small and long non-coding RNAs now. But focusing on the small RNA group, which is say 22, 24 or 25 nucleotides, here the two major players are the microRNA and the siRNA. They can be distinguished by the way they are produced, and maybe I'll talk about them in a couple of slides from, or rather immediately after this slide. What we in our group we are focusing on are the microRNA because they're coded by the endogenous genes and they regulate the different aspects of plant development under normal and stressed environment. Of course, there are some endogenous siRNAs also which are involved in the process, but then these are two distinct group of molecules. And to bring out the differences, like I said, microRNA are endogenous and siRNAs can be either endogenous or exogenous. But the major difference is that the microRNAs are coming from imperfect stem loops, whereas the siRNAs are coming from perfect stem loops. And also because microRNAs, it is an imperfect stem loop which give rise to maybe one or two molecules. They will have, here the specificity is more, the number of targets is restricted, and the regulation is much tighter, because this is the group of small RNAs that is involved in regulating the various aspects of an organism development. And also distinguishing one organism from the other. Like if we talk about monocot plants, why a rice plant looks like a rice plant and not like a maize plant. So that is regulated by the microRNAs. The molecules or the players behind the pathway may be the same, but again, the intricacies in their regulations, that is how much a protein will be formed to what level and at what time, is what is regulated. And this sets two dicot species apart from each other, and also bring about major differences when we talk about plants and animals, so on and so forth. siRNAs, on the other hand, are more arise from perfect double-stranded RNAs. They are less conserved, and since these molecules are processed, they generate a variety of different molecules. So the heterogeneity there is more, and the degree of off targets are more. And the siRNA processed transcripts can also generate, so we can have a set of primary siRNAs, secondary siRNAs, and so on. And their role is more in the defense-related pathways and in maintaining the stability of the chromosomes by silencing the transposons and other such repeat regions. So with this knowledge, let's move forward. And I'll come back to microRNAs. And I have been repeating myself saying that they are involved in regulating how how an organism is developed. So there is a variety of different microRNAs. About 8,000 molecules are known only in rice itself. And each microRNA belongs to they have multiple members to a family. That means each microRNA can arise from different loci on the chromosomes, which are, of course, now mapped thanks to the genome initiatives. And we have a lot of information on that. And most of these microRNAs were identified by genetic screens, that is by mutant phenotypes. And to show some of them here, like MIR164, which is targeting the CUC genes and have a regulation in petal number. So the mutant phenotype which was discovered was EEP1, which stands for early extrapetal 1. Here, instead of the regular 4 petals which are seen in Arabidopsis, multiple layers of petals are formed. That is, this is an abnormality because this is not normal. And hence the based on the phenotype, the mutant was named, and ultimately it was found to be associated with a non-coding RNA, which came to be known as MIR164. Now, similarly, there were other mutants like jaw, which regulates the serrated margins of the leaf in Arabidopsis plants. Then there was early flowering or the toe mutants, and then there were the MEN1 or the the flowers which had only the anthers and no pistils were present in this. So different. There are several other examples which can be quoted. So these were the classical studies were done in Arabidopsis because it was a model plant, could be worked out easily, and later other plants like rice, populars, cotton came into picture. And now we have information of microRNAs from about 70 different plants. The conserved microRNAs, that is ones which are present across different plant phenotypes, as well as the non-conserved phenotypes, which are specific to a particular plant or a particular group of plants. Having said that, another point that we need to consider when we are planning to understand or study microRNA is that although we keep on saying that they are highly conserved means they are present across all organisms, in plants as well as in animals. But plant microRNAs are very different in behavior from those of the animal microRNAs. And the major difference lies at the biogenesis step and at the function step. So in this slide, I show the difference at the biogenesis or the step of origin. Now, if there is a gene which makes, I'll talk about the animal scenario first. So there is a gene which codes for a transcript, which is known as the primary microRNA. And RNA, although single-stranded, is never linear. It never stays in the cell in a linear form. It is highly folded and acquires a complex structure. And within that complex structure, there would be a stem loop, which is recognized by a specific set of enzymes and processed to generate a hairpin stem loop, which is known as the precursor microRNA. Now, this step occurs in the nucleus, and then the precursor is exported into the cytoplasm, where it gets associated with another set of proteins to form the functional molecule or the RNA-induced silencing complex, which then performs the silencing. But this is what happens in animals. But if we look at plants where a higher level of stringency is required, the entire processing from the primary microRNA to the mature molecule is localized within the nucleus. Unlike the animals, the precursor is not exported, but the precursor also gets processed within the nucleus to form the mature molecule. The mature molecule associates with at least the AGO and is exported to the cytoplasm, where it forms the functional risks and perform the functions. And at the functional level, the two major differences is that in animals, mainly translational repression is seen. That is, the transcripts are not cleaved, but there is an obstruction in the process of translation. Some steric hindrances are created so that the protein is not involved. Whereas in plants, we have both methods or two methods of silencing at the post-transcriptional level. One is repression, and other is that the transcripts are recognized and cleaved so that they are permanently removed from the cell and are no longer available to bring about translation. In addition to this, there are some other alternative pathways which are present both in animals and in plants, which can also give rise to the microRNA. So they are the midtrons and they are the tRNA-induced small RNAs, but we'll not get into those at the moment. So the clarification at this point, this is important, that the mature molecule is exported in case of the plants, whereas the precursor is exported in case of animals, because this forms This is important for both the wet lab scientists who want to plan their experiments, as well as all the computational experts or the bioinformatic people who want to design their algorithms to predict, identify the microRNAs and their targets. And this is the major mistake normally that people do using the plant algorithms for working on animal systems and vice versa, because these algorithms will give you some results, and sometimes the results contain a lot of noise, and it gets really hard for the wet lab scientists to go ahead and validate them. Okay, so with now that we know that how microRNAs are produced and where they come from, the question is, what do they do? And if we talk about plants, then they control almost every aspect of plant biology, right from germination to different aspects of growth and development, as well as response to stress. In fact, most of the proteins that are involved in the biogenesis pathway are themselves regulated by the microRNAs due to creation of these complex feedback kind of loops. And they are also regulating many kinases and receptors involved in the hormonal signaling pathway. So each and every aspect of the plant biology seems to be under the control of these microRNAs. So more than 50%, that regulates more than 50% of the cellular gene expression. And how do they do it? At least for this 50% is by regulating the transcription factors, because each transcription factor is regulating the expression of hundreds of downstream genes. So the microRNAs can target the transcripts coding for these transcription factors, but microRNAs themselves are getting transcribed by endogenous genes, which in turn are regulating by these transcription factors. So this is the first layer or the kind of loop, regulatory loop, which is formed with the transcription factor and the microRNA regulating each other and creating gradients within the plants, and these gradients are then responsible for a transition of the plants from one stage of their life cycle to another. But in addition to that transcription factors, there are several other genes that are regulated by the microRNA. And the second layer of complexity can be seen by this cartoon, where I show how microRNAs are acting as regulatory nodes for signaling in response to different hormones. So the 5 different plant hormones I show here, the classical ones, the auxins, gibberellins, ABA, ethylene, and so on. And if you see, the auxin is regulating almost 1, 2, 3, 4, 5 microRNA hubs. Some of these are cross-regulated by the ABAs, ethylenes, and the GA. And this is what we see in nature when the plants are growing, because auxins are responsible for the growth of the plant. Classically, GA, we say, is regulating the flowers, and ABA is the stress hormone. But it is the same set of microRNAs which are involved downstream to these hormonal signalings, because what happens when the conditions are normal, a plant grows normally, produces flowers at a set time, given time, and flowers, and we harvest the fruit or the seed as per the plant in question. But whenever stress comes, ABA comes into the picture, the growth of the plant comes down, flowers are either produced early or not produced at all, and ultimately there is a loss in yield. So again, the same set of downstream genes are involved, and the regulatory hubs also appear to be common, which are occurring. And this is what we are trying to understand to see how it can be regulated. And just to make this point a little more clear, I show here in this slide how a leaf arises from the SAM, the shoot apical meristers. We all know the dome-shaped structure, which gives out the initiation, the leaf initiation, from which the leaf develops. Then the 2 domains are established. A juvenile leaf is formed, which in some cases is very different from how a mature leaf looks like. And then the plant slowly grows the leaf slowly grows, becomes mature, and then after a certain time of life, once its life cycle is over, would senesce and die. But each of these steps is regulated by several different microRNAs and their targets, which are written here. We don't need to go into each name or need to remember this, but this is just to explain the complexity of the phenomena. And so follow with me the bottom panel. We'll first talk about the middle panel. In the middle panel here, I show a Arabidopsis plant in its vegetative stage, the classical rosette. And we know that, and this is not to scale, please. This is just for explanation. A juvenile leaf is formed, grows, matures, and senesces and dies over a particular time frame. And what happens is SPL is the transcription factor which is involved in this development of the leaf, and SPL is regulated by MIR 156. So in the SAM, the levels of MIR 156 are very high, and slowly over the temporal time phase, the levels of MIR 156 start gradually come down, and since MIR is coming up, the negative regulation goes away, so the SPL levels start going up, and this is what will regulate how and when a leaf will form and develop and in the Arabidopsis plant. Now, what happens if we overexpress SPL transcription factor? This can be done easily through transgenics. Now, SPL is overexpressed, so it comes into the plant life cycle very early, and we can see here the leaf formation is terribly enhanced. The mature phase is almost abolished. The leaves form quickly and die quickly, and the rosette is very weak or poorly developed. Using a reverse scenario, where we now overexpress the MIR, which would keep the levels of SPL down so that the gradients are not produced. So here you see that MIR 156 is continuously produced, and the SPL is constantly kept at a lower level, is not allowed to rise. In this case, because of the action of the other players in the pathway, juvenile leaves are produced, and the plant maintains the juvenile leaves in that stage for a very long time. The phase transition doesn't happen, and senescence is delayed. So this is what is the power of the microRNAs. They are creating, not alone, but in coordination with their other sister molecules, are creating gradients within the plant, which will, excuse me. So they are creating gradient within the plants so that the leaf formation or the plant development can be controlled. And this is the case of Arabidopsis, which will be very different from that of a field crop like maize or rice or any other plant. Okay, so moving forward, what happens under salt stress? This is something which intrigued us, and the crop we planned to work on was, or rather, we chose to work on was rice, because rice is highly susceptible to salt stress in the early seedling stage and also at the time of seed set. And we picked up the classical Pusa basmati variety because it is the most susceptible to salt stress. And when we searched for the contrary variety, we found a variety called Pokali, which is endemic to Kerala. This is very different from the Pusa basmati background because the seeds are short and thick, but the plants are considerably tolerant and can go in the backwaters, which are slightly saline, where mixed kind of scenarios. So now we have a susceptible variety, and we could lay our hands on a tolerant variety, but the genetic backgrounds were different, and the genome is 99% similar. So how do we settle for the variance? And to our rescue came these transgenic plants, which have been generated by Professor Sapori's group. These are the glyoxylase transgenics generated in Pusa basmati background. background, where they have overexpressed two genes, glycylase 1 and glycylase 2, which convert a methyl or which detoxify a compound called methyl glycol that accumulates in the plants under stress. So we had now we could have the appropriate salt tolerant Pusa basmati control, even though it was a tolerant variety. And since we started almost as early as 2004, we we did not have too much information on rice at that time. So we started the classical way by doing a lot of NGS or at the small RNA level, at the transcriptome level, and also did a lot of degradome sequencing. In degradome is a specific kind of sequencing where the cut or the miRNA cleaved transcripts are specifically sequenced. So this would give us an idea on what are the targets and which targets are actually being cleaved in that given tissue. So again, took help of the computational biologist to do a lot of analysis for us, build the heat maps, and give us how these microRNAs levels are changing between the plants. So just as a quick overview, if the first bar here represents the microRNA profile seen in the Pusa basmati salt stressed plants, and this is where the 0% line lies. And if we see that there is a larger number of population which is getting down regulated as compared to the up regulations, and these various colors are there to represent the various fold regulations that are happening. So we are not getting into the fold levels at this level. What we need to see is that what the take home here is that majorly down regulations happening under salt stress with some up regulations happening. And on the contrary, if we look at what is happening in Pokali, this is there is a smaller level of down regulation happening as compared to the microRNAs that are getting upregulated. So the question here was, are these upregulations responsible for the tolerant phenotypes? So to compare this, we looked at the transgenic plants, and this is what we see here, that yes, and the answer is actually yes, because again, we have a larger number of upregulations as compared to the down regulations, but the fold deregulations are different. If you can see, the color bars are different, but that is understandable because in these transgenics, only one biochemical pathway was overexpressed or manipulated to make the plant's tolerance towards stress. So the transgenics in terms of MIR profile were closer to the Pokali background as compared to the regular Pusa basmati background. So this gave us an indication that we are on the right track. We then sifted through the molecules trying to see which are the ones which are getting upregulated. And by doing a lot of comparisons of salt stressed versus normal and across glycylase salt stress with the salt stress Pusa basmati, that is the different backgrounds, we could identify molecules which are showing a big jump in the deregulation patterns. We picked them up and started doing a detailed qPCR analysis of the microRNAs and their targets across different tissues to map how these profiles are coming or are behaving between these plants. And based on this analysis, we could identify a specific set or a specific subset of microRNAs which were most salt responsive and were showing, and their expression patterns were showing a negative regulation with those of their targets. And if we look at the targets, we see that these targets could be classified into three major groups. These included the transcription factors, the various enzymes, and they also included a lot of these channel transporters and signal transducers. So based on the targets of these microRNAs that came up, we could build a map which showed that the regulation occurred either within the nucleus where the gene expression is regulated, at the level of calcium signaling, which involve a lot of the signaling molecules as well as the transporters, as well as as well as the ROS pathway enzymes are also involved here. So with this information, and most of this information is now published and is accessible through papers available online. So taking all this information, we not only did Pokali and Pusa Basmati, we did several other rice varieties. We have worked on almost 9 different indica rice varieties, different tissues, different stress physiology, salt, drought, heat, combinations of two and different stages of the plant development, the early seedling stage, the mature stage, at the flowering stage, so on and so forth. So a lot of data was generated, which was compiled as a web resource. This is called ARMOR, for a rice microRNA mRNA interaction resource, and this is available for access at on the ICGB web servers. Because this information is, although is specific for rice in the Indian context, it can be used for several other uses also, because it contains a lot of information on the conserved microRNAs, which can then be searched in sister plants like sugarcane or sorghum, etc. The miRNA homologues can be identified from other plants which have not been screened so far. The knowledge about the crosstalk nodes, if a specific crosstalk node is there in your mind, can be searched. And then, of course, we have molecules which could be taken forward to for functional analysis. And I'll just give you in another 2 slides, give you a brief flavor of what we did beyond this pathway. So we took the microRNAs, we looked for how the microRNAs are distributed between the root and the shoot tissues in under normal and saline conditions, because salinity first hits the root, but the response is communicated to the shoots. And what we observed was that in Pokali, whatever Pokali grows in presence of salt. So salt is not a stress for Pokali, but if you stress it further, what happens? So whatever set of molecules are coming up in the Pokali roots, all these red dots, are actually getting downregulated under the saline conditions. So based on this analysis, we could identify actually the microRNAs and the targets like 164 NAC or 177 SCL, so on, which are operating in the roots, where the microRNAs are either upregulated and downregulated, and are regulating these transcription factors to modulate the root architecture of the plants growing under salt conditions. And these included molecules which were getting upregulated, downregulated, or behaving in a different manner. So based on these enzymes, we could identify at least 5 different molecules which were showing some very interesting behavior. And for one of the molecule turned out to be an endogenous rice molecule or a rice specific molecule, which could target the methyl transferase, the aquaporins, NAC, and a monocopper oxidase, which is involved in photosynthesis. So very diverse set of targets, regulated by a single microRNA. Epigenetic modifications are for stable transfer to them, probably to the next generation. Aquaporins are involved in water transport. NAC is regulating flower development and root architecture. Monocopper oxidase is involved in photosynthesis. So different aspects of the plant life getting regulated. We then studied how these the microRNA target pairs are behaving in the different tissues of the rice plants, even in the pre-pollinated and the post-pollinated stage. And we could observe a negative correlation coming up, where this particular microRNA turned out to be salt responsive. And not only was it salt responsive, it came in 2 forms, the 21 and 24 nucleotide forms. I'll not get into details of this, because again, another concept needs to be explained before I get here. But what we can see is that this microRNA comes in 2 forms, and these 2 forms have different targets and different modes of action within the plant. So we picked this up for our downstream analysis, generated an artificial microRNA, validated it in using in planta assays to see that whatever molecule we had engineered is cutting off the correct molecule or not. This was checked, cloned, sequenced, target cutting assays, etc. were done, and eventually we raised transgenics in the rice background. And if you can see the plants, you see that there is a certain set of plants which are more robust as compared to the wild type plants. So the phenotype was very striking. And this is how the mature plant looks. The mature plant was more robust, had a larger number of tillers. Even the root morphology was very vigorous. It had a dense panicle, which was really filled. The seed side was long. And when we grew these plants under salt stress, they resembled the wild type controlled plants which were growing. So so we did not get the seeds loss that happens in the wild type plants under salt stress is kind of compensated by these salt tolerant microRNA overexpressing lines. And based on this, we were granted a patent for it, and an international patent also is in the process. And because we plan to take this forth. And with this, I'll just, although we are proceeding with the transgenics, try to understand the mechanism so that we can also adopt the genome editing approach here. A lot of work is being progressed and done. And with this, I'll close. And before closing, I would like to thank the team of people who have been the past as well as the present members of our group. Because of them only I'm here standing before you and speaking, and of course the funding agencies. Thank you, and I'll be happy to take any questions.
Thank you,
Thank you,
madam.
ma'am. Thank you so much, ma'am, to give us sound knowledge about microRNA and their use in different stress conditions. We shall now open the floor for questions and interactions. Participants are requested to kindly raise their hands virtually or post their question in the chat box. Please keep your question brief and specific.
Professor
Ajay,
Porche,
can I?
do you have any question?
Yes.
Sir. Okay, yeah, you can ask.
Please, after you. Dr. Karthik, please go ahead. Dr. Karthik, after you.
No, I'm just asking if Professor Porche do having any question, because he worked with the genes, a lot of
Right.
genes, isomerase and all. So I hope he can interact. Professor
No,
Porche?
I just want to congratulate for this activity, and the presentation was
Thank you so
very
much.
exhaustive. Thank you very much. Great perspectives, and Karthik, you may recall that people was working on mRNAs at a school, at PhD school time you were there. So we are familiar with those things, but not we didn't reach such a level. Congratulations.
Thank you so
Okay,
much.
thanks. Ma'am, I have one question. Sir, DK sir, if you have, please ask your question.
Yeah, Dr. Neeti, the great knowledge, and you reminded us molecular biology in toto. Thank you, madam. Madam, I was thinking that, am I audible?
Yes.
Yeah, yes.
Yeah, madam, I was thinking that somewhere you defined that small RNA, the size of this RNA is 21 to 24 base pair. My question is, are they palindromic in nature or simply tandem repeats?
Okay, so microRNAs are not repeats. They are not repeats of any kind. They can be a RNA sequence of a specific specific RNA sequence. It is not necessarily a repeat sequence. It is just
Terminal inverted repeat. Yeah.
There is no repeat here.
There is no
A
repeat.
gene.
Yeah.
It is a gene, or if you look at a transcript, that transcript is processed to give you two helix structure. There
Okay.
will not be, they normally don't have a start and a stop codon. So they normally don't code for proteins, although there are one or two reports of small peptides and other things that's going in a different direction. So no, they are non-coding and they bind to the transcripts based on sequence complementarity. They may arise from repeat regions, but they are not necessarily having repeats. They can come from intron region, they can come from the exonic regions,
Okay.
from the intron-exon overlap, from the
Right.
epigenic regions, from any
Right.
region within the genome which is capable of coding.
Right.
So the so-called junk DNA is actually the portion which is the progenitor for these small RNAs of different categories.
Right, right. So my question was related with this, either exon or intron, you already defined in that way. Madam, one more thinking that came in my mind. You discussed about post-transcriptional gene regulations. Do you try to take scientific clue on transcriptional unit as well? Not post-transcriptional, rather transcriptional regulations. Have you tried or post-translational modifications as well?
Yes, yes. So like I said that there are different size categories of these microRNAs, 21, 22, 23, 24. Each size category has a different role. I'll need maybe another 15, 20 minutes to get into that, but I'm not getting into that. So there is a specific size category which which goes and binds to the freshly transcribed mRNA, that is the messenger RNA.
Right.
When it binds, then depending upon which microRNA is there, either it will get cleaved, so the transcript get cleaves in between between the exonic region, so that one half containing the 5' cap and the other half containing the poly A tail are generated, and
Right.
these transcripts are not fit to code for any protein, and hence silencing occurs, protein is not formed.
Okay.
Other methodologies, if we remember our classical molecular biology, whenever translation occurs, a sort of a loop is formed.
Yeah.
The cap and the tail comes together
Right.
as ribosomes assemble, and then the translation happen. So sometimes miRNAs go and bind, and they sterically inhibit the assembly of these ribosomes. So then also translation will not happen. Now, if the plant or the organism wants to shut off a gene expression for a short while, say under stress or under a particular condition or under response to a particular hormone, it decides that for some small amount of time, whether it could be few hours or 1 or 2 days, it doesn't want this protein to be formed.
Right.
It will undergo cleavage. It will undergo repression.
Okay.
The transcripts will just be sterically inhibited and removed from the system so that the protein is not formed. But if it wants the protein to be away from the system for a longer time, say for a week or a month or maybe over a developmental stage, then it will go and cleave the transcript so that the cleaved transcript will be recognized as aberrant. The exonucleases and will start acting on it, will remove it from the system, and again no protein will form. So in the first case, there is a possibility that the transcripts can be released back into the system so that a protein comes away. But in the second case, they are totally removed from the system so that no transcript comes. And if they want it at a later stage in development, then at that stage, microRNAs are not produced. Only the transcripts are produced so that the protein can be formed. Then there is a third instance where there is a specific information. If you have heard, there are reports on stress priming and stress memory and, different memory kinds of responses which need to be passed on to the next generation, or even in case of, perennial plants, if they need to hold that information, in that case, an imprinting or a genetic modification will happen at the genome level, which is called transcriptional gene silencing, and that region will be heterochromatized and will not be able to produce produce that protein, or the gene the transcription will not occur at that loci. So that is the transcriptional gene silencing, or a stable kind of gene silencing that will happen. So there are different modes of actions. There are different modes of how these molecules will happen, and cell has a mechanism to recognize how and when which RNA is going to do what. It is not arbitrary. So, and this distinguishing is based on the size. So 21 nucleotide has one kind of function and 24 nucleotide has another kind of a function. They will associate with a specific set of enzymes, which will lead it to perform either transcriptional silencing or post-transcriptional silencing or post-transcriptional cleavage, so on and so forth.
Okay, one more thing, Dr. Karthik, if you permit me.
Yeah, please.
Madam, as a great molecular microbiologist or molecular biologist, you are dealing over there on RNAi. Do you think sometimes that default cell machinery, cell is a chemical and coding device, we all know that. A molecular Caesar, default molecular Caesars play important role sometimes in gene silencing? What kind of view that you do have on this?
What do you mean by default molecular scissors?
Even the restriction enzymes in the cell
Okay.
cytoplasm.
No,
Right.
no, because restriction enzymes have a different mode of action. They will recognize a palindrome sequence and then they are going to make either a staggered or a blunt-ended cut.
Right.
In case of the RNAi machinery, we have two kinds of scissors. These are the dicers and the AGOs. Dicers will recognize a double-stranded hairpin loop structure and will make a cut, generating a 2-nucleotide 3' overhang. This is a specific way in which they will cut, and this cut can happen only by the dicers or the DCLs, which are involved in the first step of the small RNA generation. The AGOs will make a different kind of cut once the small RNA bind, because they are more of a functional part of the small RNAs. So when the small RNAs go and bind to the transcripts, the AGO will make a cut at the minor groove. Okay, so that the transcript and the microRNA pair is cut at the minor groove, so that two strands can be generated, and the mRNA is not functional for making a protein. So you are right in saying that they are also molecular scissors. But now, earlier we knew about only one set of molecular scissors, which were the restriction enzymes. What I'm adding onto it is an information of two more molecular scissors, but these scissors function very differently as compared to the battery of restriction enzymes that we have.
Ma'am, I
Thank
have
you.
a
Thank
question.
you.
For example, apart from silencing, microRNA also under some condition, they also induce the gene expression, some of the gene expression. So, for example, in case of salinity tolerance, whether it is possible to have a constitutive expression of some of the important microRNA that regulate regulation of genes like aquaporin, for example, or high potassium transporter, other genes, which are playing a major role in salinity tolerance. So I want to know whether we can have expression gene of these genes by mRNA apart from silencing. So what is this?
See, it is simple. I'll answer the second part of your question first. MicroRNA is going and silencing a gene. Okay.
Yeah.
And it is identifying, supposing this gene is an aquaporin. Now it is identifying the aquaporin based on sequence complementarity.
Yes.
There are two ways of preventing this. Either you change the microRNA or you change the target binding site so that it is not able to bind to the aquaporin and you can get upregulation. Now, your first question is whether whether there can be a constitutive expression.
Constitutive, yeah, apart from induced.
The transgenic plants that I showed towards the end of my talk, they are constitutively expressing the microRNA.
No, transgenic approach is okay. No? Apart from this, is there any possibility naturally through gene editing kind of things?
Yes, I didn't talk about that work in detail. I just showed you a glimpse. Yes, we are doing gene editing now to modify the expression of this microRNA. The strategies are there. See, these are part of the genome.
Yes.
It's a microRNA. Gene is a part of the genome, just like any other gene. And it is controlled by RNA polymerase II, again, just like all other genes. So we can design strategies to either edit or manipulate the expression in some way so that it comes out. Why genome editing is not getting successful or popular for microRNAs because microRNAs form gradients within the plants. MicroRNAs are produced also in gradients
Gradients, yeah.
over the temporal life cycle. But if you are editing and knocking it out, then you are knocking it out all over the plant throughout. So then the gradient is disturbed. So the
There is
strategy,
a need of some threshold.
yes, strategy cannot be a direct editing knockout based knockout strategy. It has to be a different strategy. But yes, those strategies are available and are
It
being
is possible
exploited to
because
edit.
some of the traits like salinity tolerance, if we do like that, it will be very, I mean,
Absolutely,
major
yes.
threat to the world. So
Yes.
that's okay. My audience, do you have any questions? So apart from this, the transposable elements, for example, miniature inverted, apart from normal DNA transposon, there is a category miniature inverted repeat elements, mites.
Yeah.
There are hypotheses, I mean, miRNA originated from mites like that. So did you work on this aspect with, I mean, evolutionary role also, apart from...
No, we haven't looked at the evolutionary role. We did do a collaborative work with a group in Australia on looking at the evolutionary
Because there
aspect
are
of
a lot of
microRNA.
hypotheses, but it's not yet proven. If you look at the complementary terminal inverted repeat and all, it is, we can correlate
Yes,
directly
but
with the mites
those
and miRNA.
mites are normally generating a specific category of small RNAs which get categorized under siRNA.
Okay.
MicroRNAs are slightly different.
Different,
Yes,
okay.
there are repeat-associated microRNAs which are present, and their expression varies from plant to plant. And we have a report published with Professor Robert Henry's group, where we have shown that evolutionarily, yes, they are.
Okay. Thank you so much. Thank you. Very impressive, very in-depth presentation of microRNA, siRNA silencing. Dr. Ajik, can you please conclude?
Yes. Thank you, ma'am, for patiently addressing the questions and providing such valuable insights. We deeply appreciate your time and contribution. Thank you, ma'am. Thank you so much.
Thank you.
So much. Thank you so much.
So I can exit now, right?
Yes, ma'am.
Yes.
Thank you so much.
Sure. Thank you.
Yeah. Rajay can start with
Dr. Akash.
Yeah.
Yes, it is my pleasure to introduce our speaker, Dr. Akash Shah, Assistant Professor in the Department of Computer Science and Engineering at the MIT Institute of Information Technology, MIT University, Uttar Pradesh, Noida. Dr. Shah specializes in cyber security, quantum computing, artificial intelligence, and emerging digital technologies. His research focuses on the application of artificial intelligence in climate resilient agriculture, including intelligent crop monitoring systems, predictive analytics, precision farming, and sustainable farm management. A dedicated academician and researcher, he has published several research papers, received the Best Paper Award at an international conference, and actively contributes to interdisciplinary technological innovation. We are delighted to welcome him today sessions. Dr. Shah, I kindly invite you to share your insights with the audience.
Over to you, Dr. Shah.
Thank you so much for giving me this opportunity to deliver the lecture. And thank you very much for this wonderful introduction. Yes. So my name is Dr. Akash Shah. So I would like to give you the brief overview of how the climate change can be integrated with the AI to make it more better. I can share my screen. Is my screen visible?
Yes, sir.
And I'm audible, right?
Yes, sir. Yes.
Okay. A very good afternoon. I would like to thank the MIT Institute of Organic Agriculture for organizing this FDP on the topic climate resilient agriculture and allied technologies for net zero and social inclusive framework. So, So this would be a topic, AI for climate resilient agriculture towards sustainable and inclusive farming. So basically, what is climate change? Climate change is one of the most greatest challenges facing in global agriculture nowadays. Raising the temperature, irregular rainfalls, flood, drought, pest outbreak, direct impact on crop productivity. These all are some of the reason why these climate change are affecting the agriculture. In this session, we will focus on how AI can transform the agriculture into more resilient, sustainable, and inclusive sector. So this should be my today's session overview. First, we will go with the what is climate change and how it's related to the agriculture. What is the concept of climate resilient agriculture, and what are the various roles of AI in smart farming, and various applications used in the agriculture related to the AI, sustainability and social inclusion, and lastly, challenges and the future scope. Introduction. Agriculture is highly dependent on the environmental and climatic condition, as we all know. Changes in the rainfall patterns, temperature fluctuations, and soil degradation can significantly reduce the crop yields. Traditionally, farmers relied on the experience or their intuition based on the past activity or the experience. But now, with the help of the AI, now the farmers or we can enable the evidence-based decision making through real-time data analysis and predictive modeling, which can help them for making a better decision. So basically, climate change, how it affect? Climate change affect the agriculture in multiple ways, like irregular rainfall that cause a uncertainty in crop planning. Drought reduce the water availability, while flood damages the crop and soil rising. Soil rising temperature create the heat stress for plant and livestock. Apart from this, climate change accelerates pest and disease outbreak as well. So these all are the very increasing production risks for the farmers that farmers nowadays are facing. So the concept is climate resilient agriculture. So first of all, what is climate resilient agriculture? It refers to the farming practices that can adapt to climate variability while maintaining productivity. The goal is not only to survive the climate change, but also to thrive under the changing environmental conditions. It primarily focuses on the adaptation, sustainability, and long-term food security. So this is a basic overview of climate resilient agriculture. Now, why do we need this? Today we have, I have given how the agriculture has been, farmers are facing the problem. So the need of climate resilient agriculture. There is very urgent need for this climate resilient agriculture based on climate related disasters are becoming more frequent. We see the floods, drought, and many more things are becoming very frequent nowadays. Farmer requires various strategies that maximize crop losses, stabilize the food production, improve resource efficiency, and ensure the sustainable livelihood. So this approach is essential in feeding the growing population. And nowadays, plenty of people are dependent on the agriculture, and these changes are one of the most these problems are one of the most challenging factor for the farmers. So here, this is the point of how AI can play the vital role in this climate change. AI can help the farmer to make informed decision based on the analyzing large volume of the agriculture data. That with the help of AI, the farmer can identify the patterns, predict the future risks, what are the future risks can be associated with the crop yielding or anything, and the providing the recommendation, what exactly the farmer or the person needs to do, whether it's irrigation scheduling, disease detection, yield prediction, or weather forecasting. AI will enable the AI enable the smarter farming practices. These are some of the various allied technologies with the help of AI that will help the farmer. AI work together with the several complementary technologies such as machine learning, deep learning, which are usually used to extract the pattern from the data or the data set. Some Internet of Things that are also referred as IoT. This is the device that collect the real-time information from the farms and give it to the nearby network. Drones and satellite provide the aerial monitoring, which can be in the form of video or an image. Robotics automate agriculture tasks, while cloud computing support large-scale processing and storage. So, what are the various data sources for the machine learning model to give? There are various such as weather data, the data of a weather from the last 6 years, 1 year, 2 years, 3 years, that can be given fetched to our AI model. Soil data, satellite images, drones, IoT sensor, crop and yield production. These are some of the very famous real-time dataset website. One is the most famous AI course. Plenty of people that is provided by the government of India, where various kind of a data related to the agriculture are provided, which further can be incorporated or given to the AI model, machine learning, deep learning, to give the better understanding of what particular kind of activity we need to do in the farm. Such as these are some of the various examples. That time is short, else I can share this PPT. Like I can show this as well, like this thing. These are the various of the data sets that we can easily get and can fetch to the machine learning model that can understand the pattern. So, apart from that, the first is AI-based weather forecasting. Weather forecasting is one of the most impactful applications of AI in agriculture nowadays. Weather plays a very crucial. So what AI can do, AI can analyze the historical or the real-time weather to predict the weather, the rainfall, temperature, humidity, and extreme weather events. These predictions can help the farmers to plan the planting, irrigation, and harvesting activities more effectively. AI in drought prediction. Drought can severely affect the agriculture productivity. AI system use satellite imagery, weather data, and soil moisture information to predict drought condition before they can occur. Early warning allow the farmers and policymakers to implement water conservation measures and reduce the economic loss to the farmers. Second, is AI for flood risk mapping. Flood risk mapping basically used to detect the flood-prone agricultural zones. AI can analyze the satellite and drone images to calculate the vegetation indices, detect the crop stress, monitor soil moisture, and estimate yields. This allows large-scale monitor without physically visiting each and every fold. As physically visiting each and every capturing data is very hell lot of a task. So this using the satellite images, it's become very easy. And with remote sensing in agriculture. So basically, satellite image helps us to monitor the Raj farm. And this IoT-based smart farming. So IoT, Internet of Things, we discussed earlier. It's a device that captures the data and send it to the network. So in the smart farming, IoT sensors continuously monitor soil moisture, temperature, humidity, and nutrition level of the particular soil. AI process these data into real-life information and generate the recommendation, which crop is suited for this. For example, irrigation system can automatically supply water only when needed, reducing the water wastage or improving the efficiency. So these are the various challenges. So as we see, there are various scope through which AI will help the farmer or the policymakers. But despite of these things, there are several challenges that hinder the AI adoption in agriculture. Advanced technologies can be expensive for the small farmer. Small farmer cannot use this. It's very expensive to develop an AI model to capture the data and everything. Limited digital literacy and poor internet connectivity create the barrier for the rural areas. Data privacy and security concern also attention that data, the soil data or the images will capture and go for them recognizing the pattern that can severely hinder the data privacy. Therefore, adoption must be accompanied with the capacity of building policy support. Like I will show you one basic code to how an AI model can be developed using the data set. This is the very basic. I hope this is visible. Yes. So this is the very basic code of a AI model. We basically have used machine learning model to detect analyzing the pattern to tell whether the irrigation is needed or not. So first of all, this is we call importing the libraries. Here, what we have exactly done, we imported the various libraries that we will need. First of all, the pandas, this thing. What it will do, whenever we use the data in the form of a numerical, pandas library will make it more easier for the model to understand it. Then we have sklearn, model selection, sklearn, ensemble learning, this random forest classifier. This is the name of one of the most primarily used machine learning algorithm known as random forest. We will use here to analyzing the pattern. Then this matrix, accuracy score and classification report, these are used to judge how our well model is classifying things. So this is the temporary data that we have made. This, for example purpose is temperature, humidity, soil moisture, and rainfall. Based on these features, these parameter, we will identify whether the irrigation is needed or not. Here we only have a two things, 1 and 0. 1 is for yes, 0 is for no. Suppose the temperature is 32 degree, humidity 45, soil moisture is 20, rainfall is not. So we need the irrigation as all it is. These are the data. So to first is we have used, as we see, pandas here, SPD. So first of all, we will convert all the data into the data frame. A machine learning model doesn't understand one, it basically understands the data in the form of a data frame. It's a type of a data. So these are the temperature, humidity. These are the dependent variable. Using these variable, we have to identify whether we need the irrigation or not. First of all, then we have to do the split the data set. This data set we will split into train and test part. From some model, some part of the data, the model will learn the pattern, analyzing the pattern, and then the remaining part it will test whether it will performing well, identifying whether the irrigation exactly needed or not. So this is we call train, test, and split. We have done in the form of a 0. There is 30%. 70% of the data here is used for the training purpose, and the rest 30% will be used for the testing purpose. This is how we have developed an AI model, random forest classifier. Basically, this we have done random forest classifier and fit basically give all the data to the machine learning model, X-train and Y-train. If we run this, we get this parameters. These are some of the basic default parameter that have been used here. Then, now the model gets trained over here. Now we want to test the model. Then we have to Y predict, model predict, X test. So the remaining 30% of test data will come here and it will predict whether they are performing well or not. Now we have to check the model, it's like evaluate the model, it's doing well or not. So for this, we will have two parameters for the accuracy and classification report. So here the accuracy is 1. That is means 100% accuracy. The model is very well based on the data. The accuracy is 100% is why? Because we have a very limited data. When in the real world, when we have our data in like lakhs, millions, then the data may be fluctuate. Now suppose we have some new data, new data. Now if we want to train whether this required the irrigation or not, suppose temperature 36, humidity, soil moisture, and this. Now in this we have to identify whether the irrigation is required or not. Then what we can basically do, we can call the model and fetch the new data. Here new data is this thing. So if the prediction is 0, which means recommendation is irrigation required. If the prediction is 1, which means irrigation is not required. So when we run this, the final answer irrigation is required based on these parameters. This is a one of the most basic machine learning model that I have tried to develop. So they understand how it will machine learning work in the field of the agriculture. Apart from this, there are two of the most famous applications are also there. They have integrated the concept of artificial intelligence to help the farmers or the policymakers to make it more. First is Plantex. It's a German-based mobile application where what we basically do, where people just take the picture of the crop, then by analyzing all the features, everything, it will suggest what basically disease have been associated with the crops and how can we recover. It gives a suggestion what exactly we need to do or the farmer will try to need to do. And same is one more application is crop in cloud. That does the same. It takes the picture of the land where the farmer needs to irrigate or develop the whatever farmer wants to do, and it will based on the soil data. It will identify suggest which crop will be better to irrigate at the right time. So with this, like I would like to conclude, like AI will help the agriculture in a more AI help the agriculture to make it more climate resilient, sustainable, and inclusive farming. It will help to make a better prediction, monitoring, and decision making for the farmer and the policymaker. AI will not overpass the intuition or the experience, but it will help the farmer to make a more decisive based on the decision based on the patterns or the data, factual data. AI will enable the agriculture is most important for security, net zero, and global farmer empowerment. Thank you for all giving me the attention. I really hope this session is useful and insightful for the role of AI on the climate resilient agriculture. Like I would happy if there's any answer or anything just from my side.
Dr. Ajay, yeah.
Yes, we shall now open the floor for questions and interaction. Participants are requested to kindly raise their hands
you can ask,
virtually or post their question in the
it
chat
is
box.
AI
Please keep
technology
your
related.
question brief and specific.
AI technology related. So participants, perhaps you are curious about it. You can ask some questions if you have. So I just have one question.
Yes,
Data set, large data set, small data set. Using even small data set, can we make a trained model? Can we predict something before going to use large data set? Whether it is meaningful to have a model with a small data set, how much it will be giving accuracy on that?
Like these data set are basically the sets of the data where all the past records have been maintained. But with the problem with the small data set is machine not able to fully understand, recognize the pattern. What exactly needs to done? Must be some exception. So when we use large data set, they are like 1 year, 2 year, 3 years data set, and then if he talks about in numbers, we can do about like lakhs, thousand, millions number. So data can be more, model can be more accurately trained if you have a large data set. But there's no, yes, it's better to use a large data set, but it's not any hard and fast rule that we only have to use large data set. But if we have a data constraint, like we don't have much data, then yes, machine learning model can be implemented with it. But yes, it will not give you as good result as a deep learning model can give, which is used on the, like basically what happened, machine learning basically see the analyzing the pattern. But when we have a large data set, then the concept of deep learning comes into the play. That is how it will work as a neurons. How our body, the neurons are working, same the way deep learning working. It analyzes each and every data set, data point, each and every number, and then identifying the relation between two, identifying the pattern. Like if the soil has a moisture of as much amount, what will it required, what crop will be suggested to use. It's better to use a large data set.
Large
Yes, sir.
Yeah, maybe if we don't have large data set, maybe for the test and trial, we can use small data set.
Small data set, but with the small data set, we should have used machine
for some initial kind of observation, we can
use the, yes, sir. We can use small data set.
Sir, DK sir, you have any questions?
We can see, Dr. Karthik, you can read two questions that show reflection in the chat box for Akash.
Yeah, audience, if you have any questions, you can ask.
12:35, the Nisha dropped a question. Do we have tool to know the soil characteristics? Any tool, AI tool?
So as I said, this thing, this one, crop in cloud. Basically, what happened? We just have to give the particular images of the soil or the particular longitude or latitude of the our particular area. To give it, we have the satellite. They have their own satellite. We have to do the subscription. Take the subscription. It will take the images of the data, so images of the particular soil. From the images, it will detect the what are the weather is current weather, soil moisture, how various of the features. It will take various of the feature, and based on the feature, it will suggest what are the suggestive measure that we need to take. This is one of the most very good application to use crop in cloud.
Another
Dr. I
question.
want to ask one question, sir. Just
Karthik.
like apart from script-based like Python and other program, there must be some user-friendly software kind of things. Is there for this kind of analysis, user-friendly software, or only we should go better to go with a script basis?
Basically, the application that developed by the developers are most of them are user friendly. This one, Plantix, if you like, if please use it. If you ever use it, that is very user friendly. You don't have the person only have to take the picture of a plant or a crop which have the disease. You just have to take the picture and send it to the Plantix. They will based on the picture, they will analyze what particular disease from which the plant is going on and what are suggestive measures you need to take. So basically, prior to this, this is trained over the around 2 billions of the data. The model has been trained. It will give much accurate result of very good result, and with it is very user friendly as well. So there are options for the user friendly as well.
Dr. Karthik, another question in chat box for Akash.
Yeah, please, sir. Go ahead.
Please. Akash, we
Yes.
got a message from Ravin Singh. How AI is different from the traditional methods in predicting the pattern?
So what happened in the past, people basically use the intuition or the experiences, whether the waterfall will be of how much level, whether the rain will happen or not, whether the flood will come or not, based on the experience and the intuition. But with the come of the AI, it will not only just do the, it will analyze the data. Based on the data, it will like predict the patterns. If certain things is happening, then the rain will come. If certain things is not happening, then the rain will come. So it should better have a accuracy. Nowadays, you see weather is very fluctuating. So based on all the data, like this AI will help. It will not replace the human, but it will integrate with the human mind. It will help the farmer to make it more their more profit, or like grow more crops, and which crops particularly grown in which time.
Okay. No intuition, rather we have to follow the science nowadays. Another question from Deepika Sharma. Any Indian application that can work for Indian agriculture as Plantex that is based from Berlin?
Yes, so Plantix is based from Berlin, but as of now, Indian is not a wide range of any application that is used like throughout the country. Some of the applications used from the state to state, but then they are not very much popular. So as of now, there is a need for the a software that is specifically designed for the Indian soils.
Thank you, Akash. Now, Ajay, please take over.
Okay.
Yes, sir. Thank you, sir, for patiently addressing the questions and providing such valuable insights. We deeply appreciate your time and contribution. Thank you, sir.
Thank you so much.
Thank you so much, Dr. Akash,
It was, okay, thank you.
providing a valuable
Yes.
Thank you. Yeah.
With this, we come to the end of session of first day 3 of the 5-day faculty development program on climate resilient agriculture and allied technology for net zero and socially inclusive framework. Today's session highlighted the transformative the importance of molecular and genomic approaches for developing climate-resilient crops, and the growing application of artificial intelligence and digital technologies in sustainable agricultural systems. On behalf of the Amity Institute of Organic Agriculture, I extend my heartfelt gratitude to our esteemed resource persons, Dr. Niti Sanan Misra and Dr. Akash Saha, for their enlightening presentations and valuable contributions. Thank you.
Thank you so much. Again, we can have lecture after.
Okay.
Ajay,
Yes, sir.
at 2:10, we shall meet again over here on this platform.
Okay, sir. Okay.
Yeah. So all our audience are requested, please join at 2:10.
2:10. Till then, bye-bye. Take
Bye.
lunch.
Thank you, sir.
Yeah. If Dr. Monica is there with us,
Yes, sir. Yes, sir.
Kshitij, please start.
Yes, sir.
Please.
Good afternoon,
Okay.
sir.
Okay,
I'm
sir.
there.
Okay. Thank you,
Hi,
sir.
Monica.
So, good afternoon and a very warm welcome to everyone. I am Dr. Kshitij Parmar from the Amity Institute of Organic Agriculture, Amity University, Noida, and it is my privilege to serve as the host and moderator for today's second session. On behalf of Amity Institute of Organic Agriculture, I extend a heartfelt welcome to all our notable speakers and participants. Thank you for joining us, and I hope you find today's session informative and engaging and inspiring. It is a pleasure to welcome all the distinguished resource persons, faculty members, scientists, researchers, scholars, and participants to the third day of our faculty development program. Following an engaging and enriching first session, we are delighted to continue the academic journey with the second session under the theme focusing on precision agricultural technologies and AI tool use in climate smart agriculture. This program serves as a platform for knowledge exchange, interdisciplinary learning, and meaningful discussions on innovative approaches to addressing contemporary agriculture challenges. We look forward to another day of the insightful deliberation and the fruitful interactions. To commence this session, it is my immense honor and privilege to introduce our first prominent speaker for this session, Dr. Monica Sharma, Associate Professor at the Amity Institute of Information Technology, Amity University, Uttar Pradesh. With more than 18 years of experience in teaching and research, Dr. Sharma has established herself as an expert in information security, artificial intelligence, machine learning, cloud computing, IoT, and image processing. She has published over 22 research papers and actively contributes to innovations through the patents, copyrights, workshops, and international academic engagements. Today's session focused on the transformative role of artificial intelligence in agriculture and sustainability. Dr. Sharma works on AI-enabled monitoring system, IoT-driven smart farming solutions, and data-driven decision-making, perfectly aligned with the vision of creating intelligent, climate-resilient, and the sustainable agricultural system. We are confident that her insights will inspire us to explore how emerging digital technologies can revolutionizing agriculture and contribute to a more sustainable future. So, dear all, please join me in extending a warm welcome and inviting her to deliver her expert talk. With these words, I once again extend a warm welcome to Dr. Monica Sharma and invite her to address the gathering. Over to you, ma'am. Thank you.
Well, you can
Thank you,
start.
Dr.
Thank you.
Kshitij, sir. Very nice welcome by you. First of all, I want to thank all the organizers to organize such a beautiful FDP, which is going to, it's not going to deliver the session. I have learned so many things from the past sessions too. So I want to thank everyone in this regard. Shall I share my PPT now?
Yes, ma'am.
Please, okay. Please confirm whether it's visible or not.
Yes, visible.
Okay, so good afternoon, everyone. So today's the topic of session is from soil to sustainability. As you all know, empowering agriculture with AI. So here, few things I want to discuss before starting this session. As we all understand, like what are the agriculture issues nowadays? We all know that like what are the major challenges in the agriculture area to make it sustainable. So basic issues which we have discussed means the speakers which they have discussed like the climate change, soil degradation, water scarcity, economic inequality among farmers. So these are the four major challenges which are actually need to be focused to make the agriculture sustainable. So how these challenges need to be addressed, we can use the regenerative farming and smart irrigation system and also the supportive policies. These are the three focus areas. So moving to the sustainable challenges, soil sustainable challenges are nothing but if we considering India or the other countries, so there are various challenges. Specifically, half of the cultivable land is degraded and deficient in the organic carbon. So healthy soil is essential for the food security as well as the climate resilience and the long-term farming viability. So basic and the major challenges which specifically these researchers and the developers, or we can say AI experts, need to follow or should follow. These are the major challenges like soil degradation. As we all know, like 120 million, 96 million to 120 million hectares of Indian soils are degraded. Why? Due to erosion, due to salinity, due to nutrients depletion, and even loss of SOC, means the soil organic carbon, reduce fertility as well as weaken resilience against climate stress. Water stress is again a challenge because of the poor soil structure, reduced water retention regarding to insufficient irrigation and the crop vulnerabilities during dry spells. Further, climate change pressures, as we all know that temperature rise randomly, erratic rainfalls, intensity soil erosion, and the nutrients. Because of that, there is a nutrient loss. So further overuse of chemicals is again a challenge to make our soil sustainable. Heavy use of the fertilizer, pesticides going to disrupt our microbial balance, which results long-term soil health declines. Policy is again a challenge, policy in the government map. So these are the few solutions which can be provided. These are the basic solution like soil degradation impact. We know there is a lower soil fertility, reduced crop yield, higher vulnerability to climate threat. But the solution can be the organic matter we can add. We can cover crop as well as the crop rotation can be done, reduce tillage and the prevent erosion, balance nutrients and management. So these things you all know. Like if we talk about the water stress, so there is a water loss is a big issue. Inefficient irrigation is the impact of water loss. Lower crop productivity, but solution can be mulching, improve soil organic matter, micro irrigation, rainwater harvesting. We talk about the climate change pressure. So yes, impact is more erosion, nutrient loss, uncertain yield, higher greenhouse gas emission, and the solution can be the conservation agriculture, agroforestry, soil carbon sequestration, AI and data for prediction and planning. And the overuse of chemical impact is disrupted soil microbiome, long-term soil health declines, higher costs, unstable yield. And the solution can be integrated nutrients management, biofertilizer, biopesticides we can include, then soil testing, precision farming, organic and natural inputs. And if we talk about the policy and government's maps, yes, the impact is fragmented efforts. As we all know that during the farming, there's in the segments, basically the soil in the form of segments. So impact is fragmented efforts, slow adoption, missed opportunity for regeneration. And the solution can be strong soil health policies, incentives for soil sustainable practice, farmer training and extension, as well as public-private partnership. So these are the basic solutions to make our soil sustainable. But how and why there is a need of AI? So basic need for AI, why there is a need of AI in agriculture? First of all, the precision agriculture. What is the meaning of precision agriculture here? Precision agriculture means whatever the need for the soil is, like the water, the fertilizers, as per the need only, it will going to be applied to the soil using AI. So AI is providing the solution of precision agriculture. And AI is also providing the solution, the form of predictive analytics, like the farmer can anticipate or predict the soil-borne disease in advance. Means there is some kind of soil-borne disease outbreak, or there is a nutrient deficiency. It can be early predicted by using the predictive analytics features of the AI. Next is the scalable monitoring, which is again the feature of AI. AI-enabled soil sensors and the satellite data allow continuational national-wide tracking. In yesterday's session also, there was one speaker, Bhupendra Giri sir, who also speak about sensors, and there are other speakers also. Some of the speakers speak about the sensors requirement in the field of agriculture. So yes, these sensors with AI can create a boom in the field of agriculture. In case of policy support also, AI provide transparent data-driven insight to strengthen the campaign. It's the campaign introduced by the government, which is called Meri Mati Abhiyan. You can search over the net. You will find lots of details over the net. So let's discuss how AI can help in every challenge of soil to make it sustainable, how it can solve the issues. So let's first talk about the soil degradation. Like everyone means you are more familiar in this field in comparison of me, because I'm the IT person. So you all are aware, like the soil degradation, what is the soil degradation solution will be, and what are the benefits of making the soil sustainable. So you know that organic amendments or the crop rotation is a good solution to reduce the soil degradation. But how AI can help here? See, AI-driven soil mapping identifies degraded zone and recommends site-specific amendments. What does it mean? Like we can create such kind of model which can easily predict, like in specific area, which specific area there is a need of soil organic amendments. So that can be done by AI. Same for the water stress. Like water stress sustainable solution is conservation tillage, but AI powered irrigation model can optimize use of the water. Like AI can provide a solution like that, how much moisture is in the soil and actually how much water that soil need. In case of a specific crops, that like you all aware of that there are different variety of crops, and it's not like that every crop needs same amount of water or same amount of fertilizer. So AI can provide such kind of predictions in advance, like whether the soil is in need of fertilizer or need of specific need of water or not. Next is the climate pressure. Same, as you all aware, you all have some may be having the Android phones, or some may be having the iPhones or the other phones. And most of the phones, we generally got the details of the weather forecast. Like today, there will be a weather will be clear, or there will be rain. So same, the machine AI can forecast or predict in advance like whether the climate impacts on soil health or not. Same for the use of chemical overuse solution. As we all know, like the biofertilizer or the integrated pest management, these can be the solution, and the benefit can be it going to reduce the input cost as well as the supports biodiversity. So AI can help in while taking the decision whether there is a need of how much amount of fertilizer there is a need, or how much amount of pesticide there is a need in a specific area of the farming. So that can be done by using the AI. So next, for the policy gaps, if we are taking the help of AI to upgrade the policy, so we can use the AI dashboard, which can track the soil health progressively, like whatever the policies we are using for the farming, whether they are beneficial or not, and accordingly it can be updated further to make result more accurate. So this is the image which actually shows how the soil health can be monitored using the SAIL. As you must be knowing, there is a concept of drones, there is a concept of robots in the farming. Nowadays, outside the countries, they are using such kind of facilities, like AI can analyze the soil data from the sensors, through the drones and the satellites. And soil health can be monitored easily. All you must be knowing, there are the various types of soils. Soil is also having the type, and based on that soil type, whether that soil is clay, or whether that soil is sandy, or whether that soil is in need of fertilizer or in need of water. So accordingly, the soil health can be monitored using the AI. Next, in precision agriculture also, AI can transform soil sustainability in the form of precision agriculture. Like there is a precise use of water, fertilizer, as well as the pesticides. AI can also be helpful to predict drought, floods, heat waves, as well as the impact on the soil. Like I can give you an example. Some of our students, in the last semester, I taught data analytics subject to some of the students. So my idea was, I have given them idea, like create such kind of model which can easily predict the sign of floods, droughts, in advance, so that farmers can take action in advance. The student has made some kind of projects which can easily predict the situation of floods or the drought. Though this is a continuous research phase, there is lots of improvement required in the model, because nowadays, if we talk about the data, yes, data is the major part. If we are going to train the model, so data set is the major part. If we are working on the real time data, then it will be very good. We generally get the good results. And lots of government sites are also there which are providing the real time data. So that is also good for the researcher to take those data and work on those kind of models. So smart resource management can also be done by the AI. So AI is empowering the soil sustainability in the form of chemical management, also in the form of policies and decision support system. So combining of AI, data, and action will going to make the soil sustainable as well as the secure future. Here are some challenges, like while using AI, because there might be question in someone's mind, like though we are using AI, but still there may be the challenges. So how that challenges can be overcome while making our soil sustainable. So as we all know, am I audible? Anyone please confirm?
Yes, ma'am, audible.
Okay, fine. So while using AI also, the software developers or the researchers generally face such kind of issues. Like some of the engineers or the developers, they made such kind of models which is going to predict the rainfalls, though also the rising temperature and the extreme weather. But further, the challenges are the AI and predictive models often struggles to adapt to rapidly changing climate patterns. So what does it mean? Sometimes it happens that the predictions are going in a right direction. You also might have observed like weather predictions are like, okay, after 10 days there will be the weather will be good, but what happens, sometime there will be more heat or such kind of situations occur. So there is a sudden climate change condition. So that is again a challenge in sometimes while designing a model. So again, AI also provide the solution in the form of predictive weather models like hypothetical for hyperlocal forecast or the early alert and the risk predictions can be used for the crop planning. For example, suppose we are having data of past 10 years or 5 years. We have data of 5 years weather data. On the basis of that, the pattern can be easily identified. Okay, means in this coming year, these are the situations. So there might be chance that this particular duration, there might be chance this situation may occur. So early prediction can also be done by using the AI models. Okay, so next is the case of soil degradation. Again, the as impact you all know. So I'm not going to discuss that. I'm going to discuss the challenge part here. So loss of soil fertility reduce basically the long-term productivity and sustainability. So how it can be prevented by using the AI? So you, with the help of sensors on the machine learning models, we there are the some solutions which we also have created. Like I have scholars under me who are working on this area. So what they did, so we have taken the data of some crops. We have taken the data of crops as well as the fertilizers, then the drainage. We took that data and generate the model which is going to predict that after a specific crop, which crop need to be grown or we need to be sown so that our soil should not going to degrade. So how the soil can be sustainable while making the crop rotation process is, as you all aware of, crop rotation is the solution for the soil degradation. Through AI model, we can easily predict or we can easily suggest that this kind of crop can be sown based on the conditions of the soil and the rotation. Okay, next is the water scarcity. So yes, solution, AI provide the solution, like AI optimize water use based on soil moisture as well as the weather data. And in case of economic inequality, high cost of AI code tools. In yesterday's session also, one of the moderator asked this question to the speaker, like tools are too costly. This was the question by some of the speaker, some of the moderators. Yes, there are certain things like high cost of AI tools and limited subsidies widen the gap between the large agribusiness as well as the small farmers. Researchers are working in this direction also to making those tools also sustainable, so that those tools should be easily available to the farmers. Like in the previous session also, the speaker suggested few tools. There are certain open source tools which farmer can easily use. In this direction also lots of work is going on. So hope our country will, by 2047 is going to be fully smart agri-based. Last one is the biodiversity lost. Yes, challenges is sustainable practice like crop rotation and agroforestry are underutilized. Some of the farmers, they have not good knowledge, but if we talk about integrating this knowledge with the IT perspective, integrating the knowledge with AI. So that is the challenge also, but this challenge is also solved by some of the tools. Like nowadays we have Gemini, we have other AI based tools only, which generally gives lots of information regarding in this area. So yes, AI for biodiversity monitoring, like the drones and AI can detect pest early and promote biodiversity friendly practice. So these are the certain solutions using the AI. So AI driven path to make the agriculture sustainable is a big thought. Data can be collected through the sensors, but AI can analyze that data. Smart farmers can take the action, and sustainable agriculture can be the better solution for the future. So here, one term I have added here to make this lecture more interesting. Like, as we all know, AI is for predicting the things. Like, we can predict based on the past data patterns, we can predict for the future. That is nothing but the AI. But if we are just only talking about the past data is not only the solution, as we all understand and as we all know, there are sudden changes in the climate. There is a sudden changes in the things. So what will be the further solution for that? For further solution for that is the IoT, which is the Internet of Things. So what is the purpose of IoT? Adding IoT here is IoT can generate the real-time data. So if we are having the real-time data, then the prediction will be more accurate, and also preventive action can be taken. Because if we are having the past data, then yes, prediction can be done. But preventing, if we want more accurate preventive action, then yes, IoT is a big solution with the agriculture. So for taking the decision, for taking the predictive analytics, we use AI. And for real-time data generation, IoT is the better solution, which is going to create a closed loop among the farmers to make the soil sustainable. So how IoT and AI together can be helpful, let us understand that part. Like in the past, some of the speaker discuss about the few sensors. So I have also taken example here, like though we have number of sensors, there is uncountability of sensors nowadays. If I'm going to talk, there are around 300 sensors are there, and we are generally going to talk about agriculture. Yes, number of sensors are introducing nowadays. So I have taken example of few sensors here, like the soil moisture sensor, nutrients soil sensor, temperature, humidity, and rainfall. So IoT, what IoT do? Internet of Things, it's a device which actually generates the data. It generates the data, and this generated data is going to transfer over the cloud. As and when the sensor generating data, okay, this much of humidity in the soil, that data is passing over the cloud. And here AI is going to analyze, like, what is the humidity what is the humidity ratio in the soil, and as per the whether that humidity is accurate for sowing a specific seed or growing a plant or not. And accordingly, AI provide the patterns, and it generate the insight based on the predictive analytics. And further, the farmer take the action. So here, the action is not going to stop here only. Further, there is again improvement. And how this improvement happens? See, AI has predicted the things, IoT generated the data. Further, it may happen like whatever the model has predicted is not accurate as we get the result. So there might be some gap. So this gap is again further is going to pass to the AI. Again, AI will take the charge of it. Like you can see in this diagram, as per the prediction of AI, farmer is going to take the action, and after taking the action there will be result, and further this result will going to compare with the prediction, and if there is certain gap, this model will learn again, and again it will going to pass the action to the farmer, and farmer can again take the updated action. Few things which there might be in your mind, like farmers are not having specific phones, they are not having Android phones, they are not having iPhones. So how they will take help of AI? Yes, this is the good question which asked by some of the what I can say is some of the researchers in the past sessions. This is also a good question, like how the farmers will going to take the charge, how they will going to use the AI. The same question I asked from my students: create such kind of small apps which can work in an offline mode also. So if there is no internet, but farmer will get the information, what kind of crop they should go, what kind of action they should take. Because one side we are talking about use of technology, but this technology should be reached to the particular needy person like the farmers. Means we are creating technology. If it is not reaching where exactly in need, then there is no use of creating those technologies. Okay, so I have discussed this part already, like how the sensors can be helpful for soil sustainability in terms of degradation, what soil degradation or the water stress or the climate pressure or the chemical overuse or the policy gap. So I'm not giving the brief of this slide. So let's move to another slide. So here you can see the glimpses of IoT and how the IoT is collecting the data. Means the sensors. Sensors are collecting the data. There is a AI dashboard which is displaying the data over here. I can give you an example of our campus also. In our campus, there are some smart devices using nowadays specifically for the parking management and also for tracking the organic matters. So yes, IoT sensors can generate the data, live data, and this data can be easily used to analyze using the AI for recommendation of smart irrigation, pest control, as well as planting schedule, and also for the nutrients management. Okay, so combining IoT with AI can going to give the more better solution. Okay, so just by using normal traditional methods, that was the reactive, we can say the reactive steps, like the problem detected late, then action will be taken late. But if we are going to use the AI and IoT together, it will going to be a proactive approach. Proactive approach means in advance, we can easily analyze the thing by the real-time generated data. And based on the real-time data, we can take the preventive action also. So that situation will not going to worsen. Crop yield production can be increased more with the help of AI and deep learning technologies. So here, soil health management can be easily done with the help of IoT sensors, and precision irrigation can be done with the help of real-time data collection. Nutrient management can be done with the help of machine learning as well as AI-based analytical techniques. And the crop rotation can also be done with the help of AI-based analytics recommendation system. And dashboard and alerts can be used to conserve these practices. So if we want to make an impact from reactive to proactive, so we have to choose both together for real-time data, IoT, and specifically for the prediction purpose, for taking the preventive action, we can use AI to create a smarter and more sustainable future. So IoT sensors are helpful. I'm just interrupting in between. We have time, sir, so I can continue further. Otherwise, I'll try to wind up fast. 10, 30.
You can continue, ma'am. No problem.
Okay, fine. So IoT sensors can help farmers to monitor soil health. I'm just introducing few kinds of sensor which the farmers can be used, because these sensors are not too costly, can be used easily. But one more thing in addition here, as farmers are not the technical person, so it's our duty to create such kind of small device. Though if you are going to search on Amazon, you'll find some small devices are already available online. So you can check over there and can see, you will find there will be the sensors like the moisture sensor, nutrient sensors, pH sensor, temperature sensor, and also the salinity sensor, which leading to higher yield and can reduce cost and make more sustainable farming practice. And using these sensors in India, where soil variability and the water scarcity is a major challenge, these sensor can cut water use by 30% or the fertilizer cost by also by 30%. So the use of these sensors I have added here. I'm not going in much deep here. Just I want to tell few things like how these sensors can help soil. Like soil moisture sensor can ensure crop receive the right amount of water. Moisture sensor will monitor the actual moisture of the soil because as you all aware of, there are variety of soils. Specific soil may need some less moisture or some soil need more moisture. So accordingly, yes, programming can be done. Based on that programming, sensors can predict or we can say the sensor can alert, we can generate a alert message like whether a specific soil needs some more moisture, so automatically water can be pour. Like we can take a small easy example. Like in our home also, you might have observed when we were kid, we were using the tap syntax. So we were using some kind of sensor over there which says, okay, pani ki tanki bhar gayi, just stop it. So same way this sensor works. So this sensor will going to check whether how much moisture is there in the soil as per their need, and we can generate alert message, and accordingly water can be used for the irrigation purpose, and we can reduce the cost of we can reduce the amount of use of water from the waste up to 30%. Next, for the like if we want to check the nutrients level or availability in the soil, we can use the soil pH level sensor, and we can boost yield by 10%. If the soil is healthy, then obviously yield will going to enhance. And there is a NPK sensor also, which detects nitrogen as well as phosphorus and potassium level, which guides precise fertilizer applications. And we can use electric conductivity also, which indicates soil salinity, and it helps to prevent crop failure by reducing salinity related stress. Further, we can use soil oxygen and water potential to support root respiration and the nutrient uptake. So we can use some kind of sensor which can check how much greenhouse gas is emitting. See, as we all know, that it's a bigger challenge nowadays. There is 20 to 30% green gas house emission is from agriculture only. So we can use such kind of sensors, NPK sensor, which monitors that how much number of sensors we can use, which monitor the how much gas is producing by specific crops or in the agriculture, and we can take further actions to make our environment also sustainable. So these are few images of the field, like how we can use the sensors and how it can be monitored, and how dashboard can monitor the nutrients level in the soil to make the crop yield better. So the role of AI in the soil management, sustainable soil, it's a summary of overall PPT. Like we can predict, we can use predictive analytics, we can use a number of AI models to forecast soil moisture as well as the nutrient needs, and can reduce over irrigation and fertilizer misuse. We can use AI guided equipment to apply water and fertilizer only when there is a need. So we can cut the waste from 20 to 30%. We can even detect the diseases because there is a concept of image processing in IT technology. Using the image processing technology, we can take the image and we can create the models. We can make our model learn, okay, these are the diseased leaf and these are not the diseased leaf, and accordingly we can train the model, and that model can easily predict that this particular plant there may be chance of a soil-borne disease, or there might not be chance of soil-borne disease. So early action can be taken with the help of AI. And AI can also integrate soil data with weather forecast. this thing you already know. We all have phones, smartphones. There also the sensors, and there is a data over the cloud, and that data is actually retrieving. Our mobile phones are retrieving and telling us about the weather forecast. Same way, the sensor can be used in the soil. Okay, and last is long-term soil health model. Machine learning can be identify patterns of degradation and also can suggest for the corrective action. So this is again the whole summary, how the soil sensors are generating data and how the data is collecting over the cloud and how the patterns are analyzed specifically for the nutrients deficiency checking, or whether there is a need of irrigation or recommendation, or the risk alerts, moisture trends. So yes, we can cover using the AI and IoT, we can make our agriculture and the soil sustainable. Okay. So these are the few sample examples which we have done practically. We use some AI-based and deep learning-based models for specifically for the prediction purpose. This is a sample example just to show you how the sensors are taking data. In office also we tested, like took the part of soil and just checking the humidity and how much gases are producing from a specific area. Okay, this part I've already covered, so I'm not going in much deeper. How the sensors are generating data and how this data is going over the cloud, and next how is app is detecting. So this is a sample example, how app is predicting the things and then recommending about the further action using the AI and the deep learning models. Okay, so this is all about my session.
Dr. Kshitij?
Yes, over to you, sir.
Yeah.
So these are a few references which you can use, which you can read to enhance your knowledge how deep learning based framework can be used to detect the disease and yield enhancement. So we have done this work actually. It's published in the international journal. So you can go through this work and can enhance your knowledge. And we have filed for certain patents also, and working in this area in a eager way. So the participants and the researchers who are interested for the further work can make contact on my email ID. Now I'm open for the questions. Thank you for listening.
Very nice, Dr. Monica. We appreciate your efforts. What a nice blending over here. Information technology clubbing with the agriculture and try to alleviate all the issues related with the agriculture. Very nice blending, and I think you should be here with us as a faculty in AIOA, right, Dr.
Sure, sir.
Monica? Right, because AI must be integrated nowadays as a integral approach to be associated with the curricular aspect. So we need one faculty. I shall recommend your name to my director, Dr. Doily ji. Let's see what will happen in future. Very good talk, and we learned a lot that a kind of blending data we shall provide you to put in IoT or AI manner to retrieve all kind of the parameters associated with the soil, agriculture, crop, either helping us in amelioration of the crop under abiotic or the biotic stresses. Thank you, Dr. Monica. Rest other platform, please. Other colleagues.
There are some questions from chat box. Ma'am, Dr. Jimmy John asked, are all these sensors different from each other, or are they sensors that at once detect these parameters? Buying one sensor can help at once. whether
See, sensors are different, but yes, we can create such kind of devices where all the sensor will blend together, and in the form of a single device, it can work easily. For this, there are lots of sites. You can go through Amazon. Amazon is a very popular site. So you will find lots of devices are there, but
other
we can
person
create
asked
our own
this
devices
question.
also.
Can
So
these
yes,
IoT
sensors can be
sensors
work.
purchased from Amazon be
Yes,
legally
yes.
and scientifically used for agriculture?
Yes, yes, it can be.
Considering issues related to patents, data accuracy, calibration, and publication of research findings. I have one question, ma'am. With AI technology, for example, you have developed your own sensor. You should analyze, consider with the spatio-temporal modeling. Continuously, we have to monitor them. For example, at a certain point, if we do data recording, then we stop, then it may create some data which will not be aligned with the existing data points. So what do you think about temporospatial data modeling in AI?
See, here the thing is, let me tell you, if we talk about the machine learning concept and the AI or deep learning concept, generally what happen, whatever the thing we are going to make our machine learn, machine will respond that only. But for the further enhancement, like it's not like that, it's a one go process, like machine has learned everything and will respond accordingly. Sometimes, yes, there is a need of TD, temporal difference learning. It's an advancement,
It should be
reinforcement learning concept.
whatever the case, it should be continuous.
Yes, this
the
is the
existing
case of
data.
reinforcement learning. It's like learning by its own. It's like learning from our past mistakes. We can say this. Learning from the past mistake or learning from the past data. We have past data, we have current data. Still, there might be some gap. Then we can go for temporal difference learning.
Okay, thank you so much. Audience, do you have any more questions? So, DK sir, do you want to interact? Otherwise, we can conclude because other speaker is waiting. So,
Dr. Kshitij, please carry on.
Kshitij, please.
Thank you, ma'am, for patiently addressing all the questions. And
Dr. Kshitij, 1 minute. The Professor Porchedu want to ask one question. Professor Porchedu, please ask. Sorry for a minute.
Thank you. Thank you very much. Thank you for your lecture. Now, the usefulness of those devices, the new technologies, are not under discussion. My point is how and how much of those things could be inserted, could be a topic in a course of agricultural course. We had an example for mechanization. Mechanization, you can judge if the size of the machine and such kind of things, but you are not going to talk about the four-stroke engine, etc. In this case, when you are talking about soil fertility, disease detection, water needs on the soil, etc. Just 3 examples, but there are many other examples. You have mentioned all those things, you see. If you are thinking, no, my point is, we are used to talking about disease resistance, how to detect a disease, how much should be the attack of the disease before we start spraying some chemicals, etc. And now we have a number of things in different on soil, on diseases, on water, etc.
Right.
We have to insert those things. Should they be part of a separate part of teaching, or should be included in the different topics?
Can be included in a different topics. That will be better option. But yes, it can be included in the course also. So see, the thing is, like what I understand, you want to ask like specifically disease, how the disease can be controlled using the, or can be predict using the AI and
Or
machine
water
learning.
or
That
soil
is your
fertility.
question, sir. Please confirm.
So, actually, sir is asking about biotics, biotic stress like pathogen attack and all.
Okay,
You were
okay.
talking about nutrition, abiotic stress
Fine.
like
Pathogenic.
salinity.
Fine.
Is asking whether.
Okay. Yes, can be, sir. If you're talking about the disease in the other areas of the agriculture, it is possible. We can use machine learning as well as AI techniques. For that, we need data.
Yeah,
We need
however,
either it's a past data or it's a real time data. If we are having both kind of data, yes, it's possible.
we can possible.
But yes, we need all the parameters,
But
all the parameters which are in effect.
yeah, the thing is, pathogens can be emerged. Sensitive plant may be sensitive to one day, then resistant, then sensitive. This cycle will continue. May not be predictable completely because of this resistant sensitive cycling happening for the
Sir,
pathogens.
here,
Okay.
let me give an example. What we did, we took some data of leaves only. In past, what the work we did, we take data of leaves, where we take data of diseased leaves and taken data of the normal leaves. And we got trained our model in such a way so that model can predict by just observing the specific part of the leaf and can predict, okay, this leaf, there might be chance of this kind of disease to this specific plant. Or sir, you can search over the internet also. There are certain applications, certain apps are there, but they are not, obviously not the foolproof apps, but yes, there are certain applications and tools. Softwares means the people, the software developers has designed such kind of apps. You just take a click of the specific leaf and then can predict. But yes, I'm again, this is my submission that there might be possibility that is not accurate because the data is the existing data, and also there might be chance the data is not the completely required data.
Okay, ma'am. Thank you so much. Next speaker is waiting. Really, we are thankful. Dr. Sitish, please.
Okay.
Thank you.
Thank you, ma'am, for patiently addressing all the questions and for sharing your valuable presentative with our participants. Your responses have greatly enriched today's session and provided deeper insights into the subjects. With this, we came to the end of the question answer sessions. On behalf of the organizing committee and all the participants, I extend our heartfelt gratitude to Dr. Monica Sharma for her inspiring lecture and gracious interaction with us. Thank you once again, ma'am. Thank you, ma'am.
Yeah.
Thank you, sir. Thank you very much. All
Dr.
the organizing
Sitish, we
team
can
as well
introduce
as
new speaker.
moderator, especially Erniko sirs. Thank you very much. Thank you, everyone.
Thank you.
So, as we continue with the session.
Yes. You can introduce the next speaker.
Okay, sir. It is my immense pleasure to introduce our next prominent speaker, Dr. Pankaj Kumar Tyagi.
No. Oh, you have Dr. N.K. Singh. Could you please? It is changed. The program has changed slightly. Dr. N.K. Singh. Sir is online. Yeah, so Dr. N.K. Singh is.
I have joined. Can you hear me? No.
Yes, sir. We are hearing. So Dr. N.K. Singh was a director of National Institute of Plant Biotechnology, ICR. And he was a NAS fellow elected 2007. And yeah, he did a lot of work in sequencing and in rice and fruit crops, a lot of things. So he can share his valuable lecture today about how he has gone through his milestones for the discovery of various things related to genomics and genetics. With that, we are introducing Dr. N.K. Singh to present his valuable lecture. Sir, kindly go ahead and present your lecture. We are waiting to hear you. Could you please
share the screen actually?
Sir, there is a share option. Just you can click that.
Yeah, the share option is there, but
yes, sir. Share option is there in the Zoom meeting link down.
Choose what to share with app. You must site will be able to see the content.
Uh, you can uh, you can see there must be one icon.
Share Chrome tab window,
No, sir.
enlarge screen.
If you click share, then you have to open PPT before. Then it will appear immediately. First, please open PPT directly. Then you can click share.
Yeah,
Then
PPT is open.
open. Just click share, sir. There must be one option in the.
Yes, share option is there.
Yeah, just click, sir. That one.
When I click it, it is not sharing actually. It say
when you
open
click,
system setting, choose what to share with app zoomers.
sir, otherwise, could you please send us quickly to our email or WhatsApp? We can operate for you.
Okay, let me send it, that will be better. I will send on this same one.
Sometime technical issues may happen. Even you could send us PDF file also.
This is a big file.
Yes,
You
sir.
have to
As
download
PDF format,
from
you could share us.
PPT format is there. It is
Share.
sending. Yes,
Yes.
I'm sending. It will be on Google Drive.
Okay, sir. To email. Audience, please wait. Sir is facing technical issue little bit, so we can resolve in a few minutes.
I have sent the file.
Send to WhatsApp or email, sir?
Email.
Email, no? Yeah, I will check. I'm checking.
And the same, I have replied to the the link mail which you
Yes, sir.
sent.
Yes. I'm just checking. It may take a while, little bit. Few minutes we need to wait. Sir, you send PPT file or PDF, sir?
PPT.
PPT. PPT may take time due to heavy file. If- Putting stream.
Meanwhile, sir can try at his end also by putting share and so forth.
Yeah, I'm trying actually. I'm trying, but it's not working.
Yeah. Sir, if you can convert into PDF, like
I
just
will convert into
save as
PDF.
PDF. The option of save as PDF, then it will be, I think we can get it quickly.
Dr. Kshitij? Dr. Kshitij?
Yes, sir. Yes, sir.
Meanwhile, take bio note of Dr. N.K. Singh, sir, and meanwhile you
Yeah,
can
you can
introduce.
please
Yeah,
take
it will take
bio note with the
Okay.
bio note
Okay, sir.
file. Yeah,
Okay, sir.
Dr. N.K. Singh, Nagen Kumar Singh, sir, right?
Okay, sir.
yeah. We can manage. Meanwhile, you can introduce.
Right. Because he is a great scientist with us, fellow of Science Academies,
Yeah, NASA Academy Fellow, elected in 2007.
all three academies.
Yeah.
Okay, sir.
So please check with bio note, Dr. Satish.
Right.
So I have sent a PD, I have converted to PDF and sent it.
Yes, sir.
The
Yes.
file name is NK
Yes,
Singh
sir. I'm
Amity.
just waiting to receive.
Karthik, you refresh your mail. It will
Yes,
be
sir. Yes, I am trying my
possible.
best to
So
all
today
possible.
we have Dr. Nagen Singh, sir, with us, NAS fellow, elected in 2007. Dr. Nagen Singh is a globally renowned plant geneticist and molecular biologist, recognized for his pioneering contributions to the plant genomics, genetic engineering, and molecular breeding in major crop plants. Born on 15th October 1958 in Rajapur, Mau district, Uttar Pradesh, he completed he completed his BSc and MSc from BHU and earned his PhD from University of Adelaide in 1985. Thereafter, he subsequently pursued postdoctoral research at the Commonwealth Scientific and Industrial Research Organization, CSIRO, and the University of Adelaide during 1986 to 1991. Dr. Singh sir has served in several prestigious scientific positions, including scientist at Central Food Technological Research Institute, CFTRI, associate professor at Govind Ballabh Pant University of Agriculture and Technology. Sir, I would say that I am also from Pantnagar.
Oh, great.
Yeah. And besides, he acted as principal scientist at the National Research Center on Plant Biotechnology, ICAR. He later became the national professor, holding the prestigious BP Pal Chair in Genetics and Plant Breeding at IRI New Delhi. His research has significantly advanced plant genomics and molecular breeding in rice, wheat, pigeon pea, chickpea, along with rhizobium. Dr. Singh played a pioneer role in rice genome research and the application of biotechnology for crop improvement, contributing towards enhanced productivity, stress tolerance, and sustainable agriculture. An internationally acclaimed scientist, no doubt, Dr. Singh has received numerous prestigious awards, including DBT National Bioscience Award, World Technology Award for Rice Genome Work, Dr. Rafi Ahmad Kidwai Award from ICAR, Norman Borlaug Award, and Dr. O.P. Bhasin Memorial Award. He is an elected fellow of INSA, NASI, etc. And through his groundbreaking work in genomics and molecular breeding, Dr. N.K. Singh continued to inspire scientific advancements aimed at improving crop resilience, food security, and agricultural sustainability, not only in India, but at global platform. We welcome you, sir. Meanwhile, we are retrieving your presentations. Otherwise, verbally, you can guide us if you would so. Sir, I think technical glitch at your end. You are in freeze mode. So we would request you please revive yourself and revisit it. Dr. Karthik, did you retrieve the slide? Please see.
I checked, sir. I didn't get his file. I'm checking.
Hello,
Yes.
have you shared my slides?
Actually, I didn't get it, sir, until now. You can try, sir, by clicking the share option again.
Actually, I'm trying, but okay, something has come to my screen. It is sharing, but
Share option, I think. Yeah. Okay, sir. Yeah, it's coming.
Yes,
it's
sir.
coming.
You can
Okay.
proceed, sir. Yeah.
Yes, sir. We got it.
Yeah. Thank you.
Okay. Thank you. Sorry for the
No problem, sir. No,
No.
it's happened
No
sometime.
problem, sir. Please. You bless
because
us.
It's happened.
my laptop was down. Then again, it is restoring everything.
It
Maybe
is okay,
that's
sir. It is
No
okay.
problem.
Very
Sir,
good.
please give us 2 minutes more. Dr. Shitesh, please introduce, sir.
So I think, sir, maximum things are already discussed by Professor DK, sir, about the Dr. Nagen Kumar Singh, sir. So I now invite Dr. Nagen Kumar Singh, sir, to deliver his lecture and share his valuable insights with us, sir. Over to you. Please, sir.
Yes.
okay. Good. So you can all hear me.
Yes.
Yes, sir. You can start.
So I'm going to talk about the genomics assisted climate proofing of the mega varieties of rice. That was one of the topic to develop the climate resilient crops using the genomics tools. So at the outset, I would like to acknowledge, because most of the work which I'm going to present is our own work, and there is a large number of scientists who are involved in this work as associated scientists as well as project staff and students, and the major funding agencies like Indian Council of Agriculture Research and Department of Biotechnology. So this is the outline of my presentation. Oh. Disappeared again?
Sir, maybe you can.
Is it working?
Yeah, it is appear. Yeah.
Okay. So first, I'm going to give a brief intro about the value of genomics, why genomics is useful and why we need to do it. Then again, gene discovery and utilization pipeline, and there's a big program on the climate proofing of the mega varieties of rice. I will talk about that through multi-institutional network, and search for new genes from wild rice germplasm. Then how we can use that by simultaneous mapping and integration of genes from wild rice into mega varieties. So I'm just giving very brief because we have half an hour. So I will give just one or two examples of each. So to start with, let us see what is the value of genomics, why we have to invest so much funds and efforts and time in trying to sequence the genomes of the crop plants or so many other species. What is the value of sequencing? So first of all, the genome is the complete set of genetic information present in a single haploid cell of an organism, including its protoplasm. So this definition was given more than 100 years back by Hans Winkler, who was a German botanist, and still this is the definition of genome. So when we study the structure and function of genomes, that branch of science is called genomics. First of all, starting from the very basic, once we have the complete physical map of a species, so genome sequence actually presents the physical map of this genome. ACGT, the 4 bases, they are the physical realities, unlike the genetic map, which is an abstract kind of information based on the recombination frequency. So once you have the complete physical map, you can compare the genomes of different species and get valuable evolutionary and phylogenetic insights, how the species are related to each other and how far back they had common ancestors, how close or diverse they are. This is very important from many point of view. Then as we go down for the practical side, having the genome information, we can develop high density DNA fingerprint for the characterization of our germplasm resources, which enables IPR protection, so nobody steals it without paying for it. Then we can use the genome information for QTL mapping and gene discovery and allele mining. So here the genome information actually works like a GPS map, if I can give an analogy. So having a GPS in your mobile, it is very easy to find places and people. Similarly, having a genome, it is easy to find the genes which you are looking for. It will be very difficult without the genome information. Then as a breeding tool also, we can use foreground and background selection markers with the help of genome information as a selection tool. And then finally, the genome editing, which is the most modern tool of plant breeding, is that you need the genome information. You must have the genome information because you have to devise guide RNA which will have no cross homology anywhere else in the genome. So if you want to test it that whether your guide RNA is specific to the gene of interest, then you must have the complete genome information. So by blast search, you can tell that it has a very specific position. So now the genome and genomics has become central for any branch of biology, be it agriculture or medicine or microbial biotechnology. So if you see in India, very briefly, I will tell you how it started, because the genomics revolution started globally in 1990s with the Human Genome Project and Arabidopsis Genome Project. But India did not participate in those projects. They were all multinational efforts, including China, Japan, and European and American. But we were not partners, so we were a bit late in that. So we started in year 2000 by joining International Consortium for Sequencing Rice Genome, which was the first crop plant that was sequenced. So this was a big project by DBT, funded by DBT and Delhi University South Campus and our institute IRI. We were partner. We sequenced the chromosome 11, one half of chromosome 11, the green color here. And we had to actually learn everything, and our role was actually complementary. So in that case, every country has their own chromosome to sequence and meet the global standard. So this way we actually learned all the techniques to the global standards and then participated in the completion of the genome sequence. So that time the sequencing was very hard because it was clone by clone sequencing using the Sanger's dideoxy method, a capillary sequencing. So each chromosome was cloned into large DNA fragments using back clone, bacterial artificial chromosome, about 120 kilobase pairs, and then these clones were arranged into minimum tiling path based on the back fingerprint DNA and the linkage mapping of the markers. So the back fingerprints were generated, then the back clones were hybridized with the genetically mapped markers, which are the DNA sequences, or FLP map, or any kinds of DNA markers. Then we know that which back belongs to which chromosome, and in between those markers, they are based on the overlapping fingerprints of the back. So I'm not going into the detail, but it was a clone by clone sequencing. The DNA templates has to be prepared by cloning in the bacteria, and thousands of clones has to be picked, purified, and sequenced. And in 2005, the genome was published. We finished it well within the time, and this sequence is now being used still very widely and quoted more than 5,000 times. Then we came to the tomato genome project. With our experience, we joined and we took complete chromosome 5, again South Campus and our institute. And then what happened that the tomato genome is 2.5 times bigger than rice genome. And tomato genome, 80% is the repeats. Repeats are present in this gray area here near the centromeres. They are highly condensed and they are gene poor. They are not gene rich. Whereas the orange part, which is the near the end of the chromosomes, they are rich in genes. So about 20% of the genome is gene rich, which is called euchromatin, and the 80% is near the centromere is highly condensed form of heterochromatin, which is gene poor. So because of the difficulty and the cost of sequencing, again the consortium of 10 countries, but it was decided to sequence only the gene rich part, only the euchromatin part of the tomato genome. And once again, the back libraries were prepared and shared. Because of the repeats, there was difficulty that people were not getting the bags belonging to their chromosomes. Sometimes, quite often, we were getting the bags which belong to the other chromosome, which were not assigned to us. So one more step was introduced that you do back fish first. Like this is labeled with the green and labeled with red here. So one is known back and one is unknown one. So if the unknown one goes to your chromosome, then only you go ahead and sequence that, because sequencing a back costs nearly 25 lakhs rupees and a lot of efforts. So while this was going on, In about 2008, there was a revolution in genome sequencing, what we now call NGS, next generation sequencing. And in 2008, in the PAG meeting, it was decided that we'll discontinue with this strategy because it is very slow and long binding path as compared to the NGS technology, which is very fast as compared to the old technologies like a superhighway. And then, without going into too much of detail, these are the 454, Illumina, and Solid. There were 3 different platforms. All 3 are NGS. And what was the difference from the Sanger, which we were using earlier? That now you don't need to clone the genes. You just fragment them into small fragments and immobilize them on a solid support. It may be an inert bead, it may be a glass slide, and it is amplified there itself using PCR. So PCR replaced the actually cloning. So for cloning, you need to have the pure template. And in the NGS technology, the sequencing template was prepared by in situ PCR amplification of the DNA fragments. It has its own disadvantage also because the read length was very small, so difficult to assemble, but nonetheless it was very, very cheap, maybe 10 times cheaper than the Sanger sequencing, and the large amount of data was generated. And as a result, we were able to finish the entire tomato genome, not only 20%, but the complete tomato genome. And it was published in 2012 again in Nature. And each chromosome, you can see here that where the repeats are there, where genes are there, and all the elements and some biology parts of the fleshy fruit development was analyzed with the help of the genome information. So like this, the things progress, and then using NGS, we were also able to sequence the chromosome of wheat. So all these rice, tomato, and wheat were done through international consortium. And this wheat chromosome which we took was chromosome 2A. And the chromosome 2A is twice the size of the rice genome, complete rice genome. And the wheat genome size is 6 times larger than the human genome. So it became possible only because of the NGS technology and that we could finish this wheat genome. Now the third generation sequencing came, another revolution came in genome sequencing, where now PCR is also not necessary. You just take now it is called single molecule sequencing, and the template doesn't have to be amplified. You take the DNA, you isolate the DNA from any species, any tissue, plant or animal, microbe, whatever. And then that DNA is directly, you prepare a template by ligating adapters to it. It becomes circular. And then this is put inside a well, nano-sized wells, with one polymerase, and the DNTP is fluorescently labeled. So in each well, only one DNA will be there, one polymerase, and the soup of DNTPs, and then you can actually take the image of the which nucleotide is incorporated, and in the real time you can get the sequence and very long reads. So now this is called the real-time single molecule real-time sequencing. There are 2 technologies here. One is the PacBio, this is I'm depicting here. Another one is that Oxford nanopore, where the single molecule DNA is passed through a pore and the current changes actually are measured, and by that you can do the sequencing. So you can see the sequencing has come a long way, and now we are doing PacBio, Hi-Fi, and also Oxford nanopore. These 2 are the major sequencing tool which does not require either cloning or PCR amplification. So it is a great revolution. It will become much cheaper, and genome sequences are getting completed much faster now, and is much higher quality from telomere to telomere. So using this, actually we sequence the genome of a Kala Namak rice, which is a traditional rice from the Gorakhpur area, which is the GI notified rice varieties. And we finished it with a quality which is higher than what was published in Nature, like the Nipan-Bare reference genome. It has 62 gaps, but this what we assembled in very quick time has only 33 gaps. in all the 12 chromosomes of rice. So that was a bit introductory to show that how genome sequencing we started, how important it is, and what are these uses, and how now it has become more feasible and cheaper to assemble the new genomes. Still, it is quite technically quite difficult because the assembly and annotation process are quite complex. So it is easier said than done because we don't have many telomere to telomere genome coming out from India, because we have just completed jute and pigeon pea to the full completion, and this rice calanomic which I showed you, these are all just very recent things. And some other institute like NBRI has sequenced this flex genome, linseed genome, and other people are sequencing, but mostly they are draft genome, or even if they are referenced, they are not complete fully and not annotated. So with that, now how we apply these things in the breeding process, in the crop improvement, because that's the ultimate objective from the practical utilization point of view. So here I've tried to present you a gene discovery and utilizations, process flow and timeline. So we start from the trait discovery here. Actually, this is a circular value chain. If you see here, stakeholders' preferences from production to consumption. So who are the stakeholders? Not only the farmers, it are farmers and then processor, consumers, the entire society, traders, everyone is a stakeholder, and everybody has their own requirement that what kind of varieties they want. It should be nutritious, it should be tasty, it should be easy to store, it should be higher yielding, disease resistant. So different people have different criteria that what is a good variety, and then for that we have to identify the genes for those traits and incorporate into the variety. The genes are present in the germplasm. Germplasm is improved variety, traditional variety, land races, wild relatives, they all constitute germplasm. And then you have to screen that germplasm through high throughput screening, then identify a variety or a line which carries one particular trait of value. Because all the good traits are not present in one variety. In fact, that's what the aim of the breeder is, try to get genes from different sources and put into one variety which will be a super variety. Or we move towards that and we keep on developing better and better varieties by accumulation of good genes. So the gene discovery process is that once you identify a line having a trait, then you develop a mapping population, structured mapping population, then you do genotyping and phenotyping. Then you can identify which part of the genome has those genes, and then through fine mapping and validation process, you can clone the gene, and here genome plays actually very key role in here. Without the genome, the gene discovery will take decades, and with the genome it takes just 2-3 years. And once the gene is found, then you again go back in the germplasm and amplify from different varieties and see how many versions of those genes are there, how many alleles are there with that gene, and what the difference between one allele and other allele. That means how this sequence difference in the gene affects the trait. So this way, this is completed, like the trait discovery, gene discovery, and allele mining. Finally, you find a allele of a gene which has an influence on the trait, it could be yield, it could be quality, it could be resistant. So once we have that information, then we can start molecular breeding, marker-assisted breeding. And within 3 to 5 years, we can develop a new variety over the existing variety. Whereas without this information, you go conventional way, like from here you do the crossing and selection, again crossing, field evaluation, that used to take 10 to 15 years. And then you have the variety. Again, it goes back to the society, to the stakeholders. Again, if there's some improvements are needed, like the changing lifestyle, changing climate, So then you have to bring in new traits like pigeon pea. Now you don't need very tall pigeon pea, you need which can be harvested by machine. The cauliflower, you don't need huge 2 kg cauliflower, you need a small cauliflower because it's small family and things. So the societal needs, the stakeholders needs and preferences keep on changing, and the climate is changing. Now you need drought tolerance, flood tolerance, salt tolerance. So these are a dynamic process, and so is the breeding is also a dynamic process. It can be helped by the genomics information. And so this here I wanted to show that the initial variety land races, they had more genes and alleles, because at the time of domestication we lost some of the genes, and then at the time of breeding modern varieties we lost even more genes and alleles. So now in the present high yielding variety which are represent a monoculture, lot of genes have been lost, means they have been left behind. They are in the germplasm, but in the modern variety they are not there. So we have to go back to the germplasm and get it from there and then introduce it. So like the modern varieties of wheat and rice, they are not resilient to the climate, and sometimes they don't have the same quality as the old one. So we have to search the genes for the quality and resilience and then put back into high yielding background. So here you can see the example here. We targeted three, one for yield, high grade number, one for quality, and one for the salt tolerance. There are three trait discovered lines we had, but they were not variety in their own right. They were the source of the genes. Then how first we have to discover the gene and then utilize them. So I will just give you one example here only. Same thing, same principle works for every trait. So here the gene for grain yield, high grain number, we developed the line, and this is the summary of 10 years work actually. And we found a new gene on chromosome 4 on the long arm. We find map, and then we found candidate genes here. The protein kinase domain containing protein, it is 5 times higher expression in the high grain lines, and this is the responsible for the high grain number. And then we developed markers, gel-based markers, which are tightly linked to that gene, and then introduced this gene into high-yielding variety. And then we developed a chip also. This represents the genome-wide chip, so high-density genome-wide selection to recover the background, because we want to transfer the trait and then recover the background of our best variety by backcrossing. This is called marker-assisted backcross breeding. So through this, we do the foreground selection, which is red in color here, and the background is the blue. So we transfer the genes for high grade number in the red color, and then they restore the background in the blue color, and then this is a new variety. Similarly, we took 12 mega varieties and then transferred these high grain yield gene in all of them. And one example here is a Samba Masuri, very famous variety, very eating quality is good. This is the original variety. On the left, you can see the panicle is very small. By transferring that one gene, the panicle size doubles. In fact, you can see this, the number of grains is more than double here, from 200 to 500 it became. And the number of tillers was reduced slightly, but is still 15 to 20% higher yield than Samba Masuri. So this variety has been released in Karnataka now. It is close to notification. So similarly, in different backgrounds, we have done it. Then we also fine map this QTL for the salt tolerance on chromosome 7. And then that is also been used in 3-4 varieties have been released in the same way. So the time is short, so I'm not going into too much detail, but in principle, we discovered the gene, we developed the DNA marker, and through marker-assisted selection of foreground and background, we put them a gene, valuable gene in the high-yielding background. So the principle is that, we have some varieties of rice which are called mega varieties, because they are very popular. They are grown in more than 1 million hectares. These are like IR64, Swarna, Samba, Masuri, MTU 1010, Pusa 1121. These are being grown for last maybe more than 20 years, but they are still popular among the farmers. So they have lot of their rare combination of the genes and alleles, well adapted, but they lack something. They lack the climate resilience. Sometimes they lack some, traits which we like to transfer to them. So it becomes a new variety. On the other hand, the traditional varieties, they have unique quality, high level of stress tolerance, but they are low yielding, so farmers are reluctant to grow these. So then we have to, take good genes from there and put in the high yielding background. Here there are different methods of molecular breeding. For example, mass marker assisted selection and the pedigree breeding that requires large population, marker assisted recurrent selection, like you cross and then bulk again cross. This is basically for the cross pollinated crops. Marker assisted backcross breeding is the most successful one. I showed you one example and I will show you more examples of this, where you can put to backcross breeding, you take already a established variety, mega variety, and to put the genes which are lacking there. Then there are genomic selection, which is based on the genomics assisted breeding value of large number of markers, and then one can use it as a selection tool. It is more useful for the cattle breeding and the tree breeding where the population size is not that big, but you want to select the best out of that excuse me, that is small population size. But for large population size, wheat and rice, that is not this suitable thing. So the biggest challenge, this one you should take note here. The biggest challenge is that the odds are against you. Odd means the probability of, even if you take 10 genes and you want to 10 genes, they will be biallelic. If you cross the two variety, there are two alleles, one is good, one is bad. So when you cross it, then in the progeny, if you want the good alleles for all the 10 genes to come together in the progeny, the chance is 1 in 1 million. Okay, so the chance is very low because this is the formula here. 1/4 raised to the power n. Here, if you have 12 genes, that becomes 1 in 16 million, because every time you have to keep on multiplying by 4. So you know the Mendelian dihabit segregation, the checkerboard, 1 in 16. When you have only 2 genes, it is 1 in 16 will have the better allele of both the genes, and you keep on multiplying 4 times 4 times 4 times 4. When the 10 genes becomes 1 in million, 12 gene, 11 gene, 1 in 4 million, 12 gene, 1 in 16 million. And there is no shortcut to this actually. That is the shortest possible way. Suppose I have a near isogenic lines, which the background is perfect, everything is very best background, and I want to put just 4 genes together. So for putting the 4 genes together in the progeny, I have to have a population of 256, then I will get 1. But you don't want only 1, you want at least 10. So that becomes 2000. And similarly, that is only 4 genes. And as the number of genes increases, this is the biggest challenge which we have. And that is the numbers game. And using most precise tool also, it is quite difficult. But the genome editing can provide a shortcut where you can put a number of genes on the same construct. So if you put 4 genes on the same construct, then the 4 genes behave like a 1 gene. So anyway, that is the theoretical consideration, and that is a mathematics really, that the odds are stacked against us. That's why we don't get good variety every day, because the chance of getting is very low. Most breeders are playing in 2,000, 3,000, 5,000 population size, whereas the requirement is in the millions. If you want to put few good genes together, the population has to be very large. So here we developed a big program of the multi-institutional, 12 different institutes, different part of the country is a map here. And we went on in a very focused way. We took the mega variety which are popular in that region, and then tried to introduce genes for drought tolerance, flooding tolerance, and the salinity tolerance. So we took 7 genes for drought tolerance, which is DTY1, DTY2, DTY3, like this. and SALT are for salinity tolerance, and sub-1 for submergence tolerance. So these genes are QTL. QTL means you know the narrow region where the gene is, but you don't actually know what is the gene, because that requires fine mapping and cloning. So this has got Saltal, the gene is known, sub-1, the gene is known, but the drought QTLs, gene was not known, but the very precise markers are known. So that doesn't matter. You don't have to know the gene. You can do back cross breeding using the marker. So within 2-3 back crosses, we can restore the background and we have the presence of the foreground. So we developed at our institute the foreground markers, which are easily used by then passed on to breeder, and the breeder did the selection for the foregrounds, and we did the background selection using the chip, because it is very costly and little bit of technically difficult. And then you can see here we released 19 variety, and this year 3 more varieties were released. So the total 22 varieties have been released in a single project. And that is no other project has released so many varieties in a single project. And all these varieties, they are climate resilient versions of the mega variety. Mega variety means all already popular variety like Swarna, Sambhavan, MTU 1010. So there is no question of acceptance and adaptability. And those varieties are already popular still after 40 years of release. But some of the things which they don't have, those traits have been added, and new version of those varieties have been released. And they are released and notified and getting very, very popular. So, for example, one such is DRR Dhan 50, released in 2018. It has both submergence and submergence and drought tolerance. And with a single irrigation or rain-fed condition, it produces 5 tons per hectare. Whereas the original Samba Masuri will be completely dried without water. Then M2U 1293, it has high level of salt tolerance in the coastal Andhra Pradesh. It has been released and becoming very popular. Similarly, Malvi Dhan 105 sub-1, it is submergence tolerant. Now the fourth aspect I'm coming, it will not take a lot of time, I will go through. See, the genes are there in the traditional variety, but many more genes are there in the wild relatives of the crop plants, because they are growing in the wild without any protection. And they have survived in nature through millions of years up, and they have seen all the global climate change of ice age and the global warming. They have been through that, so they have accumulated the genes for stress tolerance. So I've collected wild rice, more than 1,000 wild rice from different parts of the countries. They are normally on the roadside, in the shallow ponds, in the river basins, and small ponds. They are there. They are also on the upland area from from Himalaya to the Western Ghats. In a small patch of water, they will be growing, and they just shatter there, again go, again shatter. This they are doing for millions of years, not thousands of years. And nobody is there to protect them. It has protected itself through the genetic evolution. And they also grow in the stressful area like this. This is very, very sodic soil. The white color you can see is basically salt. This is the sea water here, inundated sea water in coastal Karwar, Karnataka. Then the dry area and the complete submergence, they are adapted to all kinds of situation, and the variety which is adapted must have genes for that. But they are under threat because of the developmental pressure, widening of road, industrial parks, housing building, school building. They are all disappearing very fast. So we need to protect them. Well, I have collected about 1,000 and preserved them in NBPGR. We have analyzed them through high density fingerprint. They belong to 3 different subpopulation of Raja Rupi Pogan. Then when we screen them for different traits, like for drought tolerance, we find lines which are highly drought tolerant. We find lines which are tolerant to anaerobic conditions during germination, like flooding during germination, where most of these variety will not germinate at all, they will die. Then we found salt tolerant lines also, more tolerant than focali, and then BLB resistant line, almost immune to BLB, then sheath blight resistant, then brown spot resistance, also the nematode resistance. These things are all published well by different collaborators. You can see the basmati 1121 has a big nematode, root knot nematodes. The wild rice does not have any, it is immune. So I'll just give you only one example, is that we took one of these lines which were tolerant to salinity, 173, and then cross them and then back cross. In the back cross 1, we did the mapping. For mapping, you have to do genotyping and phenotyping. So here is the genotyping using that same chip which I described. It has 50,000 SNP. So we found about 1,000 SNP which were polymorphic, and they were segregating 1:1 as expected. And then we did phenotyping for salt tolerance. You can see here is the number 1 is tolerant and number 5 is sensitive. So it is segregating here. This is the IR64 highly sensitive. Everything is dies. And then this the wild rice survives. And this is the segregating population. Other traits are also segregating, like the black color is the wild color, on is wild trait, and you get lot of segregation for different traits, but our aim was to look for this salt tolerance only, and then we mapped it. See, on the chromosome you get very high density map because you have high density genotyping. Then we found these 5 QTLs coming from this wild rice, and they have been introgressed into IR64. Then now we have the new source of salt tolerance. Similarly, all those traits which we have identified, some more are there. I've just given you one example only. That is the pathway of utilization of our genetic resources. So this is my last summary slide. What are the challenges and what is the way forward? The most important challenge is the conservation and high throughput evaluation of the germplasm, because you have seen that how the germplasm is disappearing because of the developmental pressure. Not only rice, so many other crop species also we are losing our biodiversity very fast because of the developmental pressure. Then we have to do high resolution mapping using high density SNP arrays and identify the gene, clone them and validate them using genomic tools, then develop marker, and as I have shown you that we can utilize them through accelerated breeding process, and we can also utilize them through genome editing using SDN2. SDN2 is the tool for allele replacement. You can replace the allele, the bad allele with the good allele, and that will be not treated as a GM because it is still within the rice and at the same position. So that way you can develop new varieties. So genome editing is actually very powerful tool which will also take care of the statistical arts which we have of putting more good genes together. It is very hard, means probability is very low, but through genome editing it may enhanced. So with that I close it. Thank you for your attention. If you have any question, I will be happy to answer.
Audience, do you have any questions?
Thank you very much, sir, for your highly insightful lecture. Your valuable perspectives have greatly enriched our understanding of these important areas of research. So we shall now move to the question answer sessions. Participants are requested to post their questions in the chat box, or you can raise your hand.
Meanwhile, sir, what do you think about pan genome? Did you do any pan genome of different rice varieties as a reference?
Yes, rice pan genome is already available using the 3,000 genome panel. 3,000 varieties have been sequenced and the pan genome has been developed. So that pan genome is also very important, that because all the genes are not present in one variety.
Yeah.
Every variety will have some unique genes which is not present in other variety. So pan genome definitely represents the entire species. So as you sequence, more and more variety, you keep on adding to the existing number of genes, but it reaches a saturation point after some times. You don't get much gain, but initially you will have good gains. So single variety is not enough, but single variety is start. Like if you are working on a new species, then you have to start with the reference genome and then sequence few more genotypes, maybe 200, 300, and you can develop a pan genome. That will be a better reference than a single genome.
What about doubled haploid things?
See, double haploid again is a very powerful technology which we have not used in India. Actually, unfortunately, we must use it, but globally it is being used. Like
Yeah.
in all this, in the CG institutes, like in Mexico, in Australia, Canada, USA.
Yeah.
Double haploid technique is you can reach 100% homozygosity. That is basically to create the inbred lines, like the homozygous lines.
Yes, sir. Yes, sir.
So
Yeah.
if you do the
For potential
microspore,
variety. Yeah.
yeah. So if you do either microspore culture or the chromosomal elimination technique, there are techniques to produce haploid, and then you
Yes.
colchicine it to make it double. So the advantage is that within the single generation, we reach the complete homozygosity. There is no need to do further selfing or backcrossing. So this is actually an alternative to speed breeding. What people, there is a lot of hue and cry about the speed breeding, but
Yeah.
in my view, you don't have to do that. Double haploid is more powerful than
They're
speed breeding
powerful. Okay.
because you get all your recombination in F1. In the next generation, you get a little bit more recombination, but I think we can have more plants. But what is important is rather than selfing for 4 5 generation in a year under artificial light, the plant get stress. If possible, if you have double haploid, then go for that. And I think people are going for that.
You have showed you have collected lot of wild relatives across India. So how much they were useful to have to increase yield or resistance to biotic or abiotic stresses with our wild relatives collection from India, sir, according to our experience.
Yeah. I think this is very useful, particularly for stress tolerance. For the yield, actually, there are hidden genes are there
Yes.
because the cultivated varieties are more high yielding than wild relatives because they have so many undesirable traits like the seed scattering and uneven
Yes.
maturity.
Yes.
And but still they will have a lot of tillering. They have very high tillering as compared to any variety. So the tillering trait is there and the vigorousness is there. So they have many traits which can be introduced, but as such they are not hiding. But definitely for all the stresses, because they are under nature under the stress and they have adapted. So I showed you one example of this salt tolerance.
So yes,
But
salt
we,
tolerance. Yeah, especially from
a
natural.
very old example is this grassy stunt virus in case of rice. It used to be very big problem. A virus which will make the rice very grassy and it will not head, pancake will not come. Even the last 2 3 years there was some outbreak of that kind of virus coming from Taiwan or some other country. But it was very widespread. Then Dr. G.S. Kuss he introduced from the one of the wild rice, Oraja nivara, and then all our modern varieties are free from that virus. They are almost immune from that virus. So we
So
don't
there is
talk.
a okay, sir. Yeah.
And also these for the hybrid rice, this that is also coming from the wild rice only. The alien cytoplasm. You get
Wild relatives are really very useful.
very
The problem may be interspecific hybridization, little bit difficult, but if you overcome some kind of introgression, maybe very useful.
that is true, obviously.
So there is a question from audience. One question. Sir, which advanced genomic tool has been used to identify elite genotypes for breeding programs?
Say for breeding program, you need the trait donors.
Yes.
Trait donors can only be identified by screening for the trait, not by any genomic tools.
Yeah, screening percentage directly.
You have to screen for that particular trait. So you have to develop high throughput screening techniques. like in a small parts or in a small, test tube. If you can screen large number, then you can identify the donor of the trait. And that donor can be used in breeding.
You are saying like a forward genetic instead of the reverse genetic, directly from phenotype to genotype.
Reverse genetics is a different thing. Reverse genetics, when we know the gene sequence, but we
Gene
don't know the
sequence.
function of that gene.
Yeah.
So we create the mutations in that gene, and then we see that what function it has.
Yeah.
And forward genetics means you know the trait, but you want to identify the genes.
Identify. Yeah.
So I have just talked about that. You have to develop a segregating population and genotyping, phenotyping, then you can get to the gene.
So
But as a donor for the breeding, these techniques have no value. For selecting the donors, you have to screen for the trait. Like
screening. Yeah.
if you want to donor for the bacterial blight resistance, then you have to screen for bacterial blight. Genome will not tell you that.
Yes, and also if we go with an our conventional QTL mapping and all, maybe there are problems with the linkage drag, as you said. Maybe from the background, some other genes may unwanted genes may be expressed.
You are right. We encountered that kind of situation where, like for example, SD1 gene, which has been used for green revolution
Yes.
to make the rice dwarf, that
Yes.
is very, very tightly linked for a QTL for drought tolerance, like DTY 1.1. So in our program through ERI and Dr. Prashant, we broke that linkage. So now we have the dwarf rice, but it is drought tolerant for that particular locus. Of course, there are many loci for the drought tolerance, but that was a very important loci. So
Yeah,
that
SD1 semi-dwarf. Yeah.
yeah, that was broken, and then you need large population, and you have to do very fine mapping.
Yes.
And their genomic tools are used because you have to design primer for any narrow region, SNP markers, and then you break that unholy linkage. So that is a problem actually.
Alternatively, we can do gene knockout through genome editing. Like
Right. So that's why genome editing is a very powerful future tool.
to eliminate unwanted background genes.
It has so many advantages actually. Only disadvantage is that people might say that it is a GMO, so there will be opposition, but that has been cleared now that allele replacement is not GMO.
Genome, yeah.
Only
Native genes can be edited.
Yeah, only when you put a foreign gene like from bacteria or from other species, which normally cannot be transferred by crossing, then it is GMO. If something you can transfer by crossing anyway, this only speeds up.
Yes.
Genome editing.
Already just editing the native one.
Yeah. So two types of editing is there. One is the creating knockouts, like you have a gene and you by non-homologous end joining, you cut the DNA and while joining, there's a mutation is created. That is SDN1. And in SDN2, this is homology directed repair.
Yes.
Then you can transfer
Very
entire
powerful.
allele.
Yeah.
Entire allele, you can take a good allele and through recombination, you can direct it to the locus and then they change the allele. That is also free from regulation. That is called SDN2. So it has been defined that allele replacement within the primary gene pool will be treated as SDN2 and it will be not regulated like GMO.
Yes,
But
without off targets, etc.
we don't have good techniques for that yet. So most of the gene editing is being done by knocking out and knocking down. For allele replacement, still we have to work on and develop good tools that how neatly we can, just replace that allele through recombination. So
That
then
will
people
be
will work out, and this is only a matter of time. If you are at it, you will get it.
Okay, sir. Really, thank you so much for such an inspiring lecture from you, from the milestones of your various work. Dr. Sitish, if there is no question, sir, DK sir, please.
Yes, sir. Actually, DK sir.
Yes.
Sir, being a microbiologist, I would like to say that do you agree that horizontal gene transfer may impose some characters on the host genome for generations? under any abiotic stress, if any pathogen attacks
Yes,
to
sir.
any rice or any crop, what is your opinion? Does a microbial gene may integrate with the host genome and it carries its traits to the host genome for generations?
Yes, sir. That actually happens. We have noticed that, and when we analyze the genome, we find genes coming from all kinds of different sources. And their frequency of transfer is very low, but that happens in nature. In fact, that keeps on happening. Like you have agrobacterium genes transferred in host. You have even I was analyzing the jute genome, and we found some CRISPR genes there. The
Oh.
CRISPR-Cas is coming from bacteria,
Right,
and we are using it we are treating it as a foreign gene.
right,
When we did the complete annotation, there the CRISPR gene was there.
right,
And even some other people have also noticed, in fact, and they were alerted that is this some contamination coming because people are working on this CRISPR rice. But then when I found in jute CRISPR gene, then I say that in the nature it has been happening at a low frequency. So
right,
the microbial, which in fact, definitely because they are together, and at the time of cell division, there is a chance of
right,
integration. And the typical mitochondria and chloroplast are the typical example of that.
right,
Because
sir.
the entire microbe has been inserted, and then from within that, and there are thousands of places that chloroplast fragments are integrated into nuclear genome,
Right,
and also similarly microbial genomes.
right,
Mitochondrial is also same way. So it in the nature, and the horizontal gene transfer has played a lot of role in the evolution.
right,
And maybe sometimes some new genes which are very useful,
right.
they have got transferred and got selected. And once it is integrated in the chromosome, then it will naturally inherit generation after generation.
Right, sir. Thank you,
Thank you,
sir.
sir. Professor Enrico Porcedo from Italy want to ask you some questions. Sir, please.
Just a question. Any experience on inverse genetics? I mean, analyzing the variation, the different species and subspecies of rice, and detecting the differences between or among groups of populations.
You mean
Instead of going to the phenotype and trying to identify the gene and the alleles on that specific gene, analyzing your collections, cultivated and wild material, and detecting differences between the different groups or different species and subspecies, and exploring the variation you have on the same genus.
Yeah, that is true. I mean, there are some examples, but mostly we have to evaluate under that stress situation, and they identify the donor sources. And some natural hybridization has also taken place. In fact, in case of rice, lot of natural outcrossing with the Oryza rufipogon and Oryza nivara takes place. And in that, huge amount of variability is created. And within that, in situ, I have noticed that actually, where the cultivated and the wild are going side by side, and then on the periphery, there's hundreds of different types are generated. And there we find lot of different sources of resistance, but they are not good as a cultivar themselves. So unless we backcross them into the cultivated one, because that has too many undesirable traits for the cultivation purpose. So similarly, the related species which are naturally crossing, we don't have to go for that. We simply evaluate them as a germplasm and then make use of inbreeding. So I'm not sure exactly what you wanted to know about the inverse genetics. The reverse genetics is there where we create the mutations. We know the gene sequence, but we don't know the function. So in a scientist in Korea, he created the entire mutations through transposon mutagenesis. He almost inserted a transposon in almost every gene. and then he correlated with the phenotype, that what changes occurred in the phenotype. That was the reverse genetics for the functional genomics. But we are more interested in the natural variation in the closely related species because they are adapted to different conditions of adverse conditions. So more likely to possess the genes which have evolved over the years or over thousands of years. And some of them may be sitting silent because maybe the pressure was there at some time, but now the pressure is not there, but the gene is still there. So when we select under artificial conditions, we identify those donors of those traits. Particularly these, abiotic stresses like the heat and cold and the flooding and drought, we find a lot of variation, hidden variation.
So there are other two questions from audience. One question, Dr. Jimmy John is asking, Sir, considering our long-standing efforts in multiple stress tolerance, what direction or advice should we give to young researchers looking to make an impact in rice research?
Well, what I feel, that we have to go in a more precise way because now we have tools to go in a precise way. So we know that what are the stresses. It could be cold, it could be heat, it could be drought, it could be flood, it could be salinity. All the abiotic stresses are known.
Biotic stress.
And there are genes for that, naturally. They are genetically controlled because the lines are there which are tolerant, so they have genes. For example, we found the genes for heat tolerant also in N22, one of the land races. So we have to precisely go to the gene, identify the genes, and then transfer them using these molecular breeding tools. I don't see any other way actually. It is any shortcut is not there. And then we try to pyramid them. So we have not reached to the pyramiding stage. What I showed some examples are just one or two genes being transferred.
Yeah.
And then, so we do in parallelly, we choose a very good variety, standard variety, and then we create isogenic lines in parallel with different genes. And then we can intercross, and from the F2 you can select the gene pyramids. So we recently published one paper where we have pyramided 3 genes per drought and 1 for summer genes and 1 for that. So that is possible.
So focusing on different stresses, yield, etc.
So the background, it is, my advice is that we take the best background, because background, getting a good background itself is a challenge. So these mega varieties, say when you have this IR64, something like that, bred at Erie, this is a super variety and it has a rare combination of all the genes. Once you cross that with any variety, you disturb the apple cart, Then to get the same combination again the challenge is that the chance of getting, several good genes together is very, very low. So the back cross breeding, in my opinion, or genome editing on a base of already very superior variety is the way to go. Of course, we can look for better combinations also simultaneously, but the chance of finding is very low, simply by statistical probability. Finding something better than that, the chance is low, but adding on to that, conglomerate of all the genes, adding individual genes, discovering and adding them is more fruitful, and it will be faster because I don't see any shortcut. There is no shortcut actually, because the odds are against us. Only shortcut is that if you can put 10 good genes in a single construct and they transfer them, then that will become GMO. That will be not free
Yes,
from
that
regulation.
must be transgenic.
It will be free from regulation only if you do allele replacement at the same locus.
Yes.
So in the Indian regulation, it is defined that even if the gene is from the same species, if you transfer at a different locus, then
that can be considered as transgenic only.
That will be transgenic. That will be not free from regulation. So,
The
but
same locus.
maybe in future people will change this regulation that again, if the gene is not coming from foreign source, then let them put 10 genes together. So 10 genes behave like one gene. So our chance of putting multiple good genes together increases. It is not in millions, but it is in thousands. Otherwise, if you just put 10 good genes together in 1 million,
So,
you
I
don't
have
have
one
1 billion.
simple question. Rice is diploid. In case of wheat is polyploid, hexaploid. All genes probably have 3 versions, 3 copies.
Yeah, yeah.
Do you think maybe one is active, other 2 may not be active in the case of wheat? So my question is, when we go on gene editing, should we consider all 3 copies, or can we go with only 1 copy? What is your suggestion, sir?
See, wheat is, of course, is a hexaploid, but it behaves like a diploid.
Yes.
And in generally, most of the genes will have 3 copies, and some of maybe there's a diploidization actually happens after polyploidization. Diploidization.
Diploidization, yes.
So it could be all 3 may be active, 2 may be active, 1 may be active. So there are 2 things. If you are going for knockdown,
yes,
then you have to knock down all 3.
yeah,
But if you are going to add 1, then even single dose will have its effect.
okay.
So, for now, in a polyploid, like even in mustard, also the same thing, that if you want to make double zero, there are 2 alleles are there, both have to be knocked down. Otherwise, even one is
Yeah.
active, that
For
will be.
knocking down, it is okay, but for induction of some genes,
For
maybe
adding
3 copies if they are
traits,
working.
adding trait, one copy is enough. We don't
Yeah.
need to have 3 copies.
Yeah, one copy.
If the gene is good, I think there are many examples also. It's a polyploid, but in only one genome has the gene.
Yes.
It is useful.
Other
And
may
that-
not be active.
And that works.
Mm.
It doesn't have to be in 6 copies, to work. One good gene in only one chromosome, 1 copy is good enough.
Yes.
But for knocking down, all the copies have to be knocked down.
Okay, sir. Sir, one more question from audience, a general question about CRISPR. Sir, in most of the breeding approach, why CRISPR gene has been used extensively almost in all the crops, and it has been increasing for, whether it has been increasing for precision breeding? Is there any specific features for the gene?
So it is the discovery and development surrounding the genome.
It is a technical.
Yeah,
It's
it
a
is
technique.
technical because at the present situation, CRISPR is the best tool.
Yeah, best tool.
People are using. Maybe in future people will come with better than that. And even CRISPR, there are different versions are there.
Yeah, there are other tools also, TAL and other tools also there.
Talent,
But,
they were
yeah,
less precise actually. They were
less precision. Yeah,
they were
that is
in the
the reason
past.
CRISPR we are using this.
Presently,
That's
CRISPR is the zinc
most,
finger,
I
zinc
think most
finger
precision.
protein and talent. They actually the past technology.
Yes.
Present technology is CRISPR, and
CRISPR,
in
yes.
CRISPR also there are different versions. People are devising techniques, are getting patents over it.
Yeah, new versions of this
Yeah.
Cas9, they are mutating and creating better versions.
Right. And you may come up with completely different way of doing that targeted mutagenesis. It is in future. Who knows? You may
Yes.
find a very very short, very very simple way of doing that.
Maybe yes, like Sankar sequence to NGS sequencing now advanced. Similarly, yeah, for
Similarly,
gene editing.
don't know
also.
what is in future, what is lying in the future.
Yes.
But if we keep on thinking and thinking over it and trying different things, that is the way to get it, because it's a human mind which keeps on devising new things and say things. I'm sure you'll get better way of doing, but in today's, that is the best, and that's why everybody is using it.
Yes, correct, sir, correct, definitely. So thank you so much, sir. Dr. Sitij, please. Unmute Dr. Sitij. Dr. Sitij, please unmute.
If it does not work, Dr. Karthik, you may lead and wrap up the session.
Okay, sir. Thank you so much, sir. Really, very inspiring talk. And you have described about the cereals, fruit crops, and a lot of things about sequencing from Sankars to NGS with various platforms.
Sir, now it is working.
Yeah, okay, sir. Yeah, you can.
Okay. So, thank you so much, Dr. Singh, sir, for your enlightening presentation and for patiently responding to the questions raised by the participants. Your valuable insights and expertise have made this session both informative and inspiring. On behalf of the organizing committee and all the participants, I extend our sincere gratitude for your time and for sharing your knowledge with us. Thank you so much, sir. With this, we come to the successful conclusion of this session 2, the third day of the faculty development program. Today's
thank
session
you.
provided valuable insights into the climatic resilience agriculture and highlighted the transformative role of AI, machine learning, IoT, precision agriculture, genomics, and the molecular breeding in the building climate smart and sustainable farming system. The deliberation emphasized that technology-driven innovation combined with in advances in plant genetics and biotechnology is key to enhancing crop productivity, optimizing resource, strengthening food security, and ensuring agricultural resilience for the future. The keynote address by Dr. Nagin Kumar Singh offered a deeper understanding of how plant genomics, genetics, engineering, and the molecular breeding can contribute to the development of high-yielding, stress-tolerant, and climate-resilient crop varieties. His pioneering work in the rice genomic research and crop improvement has significantly advanced sustainable agricultural development and reinforced the importance of integrating biotechnology with modern farming practices. I am confident that the knowledge and experience shared today will inspire new ideas, collaboration, and research directions among all participants. On behalf of the organizing committee and our institution, I extend our heartfelt gratitude to our esteemed speakers, Dr. Monika Sharma and Dr. Nagin Kumar Singh, for their enlightening lectures and for the sharing their valuable expertise and experience with us. I would also like to thank all the participants for their enthusiastic participation, insightful questions, and active engagement throughout the day. Thank you so much, sir. Thank you all.
Thank you, sir. Really, thank you so much.
Thank you, Dr.
Giving
Karthik.
such a lecture in your busy schedule. Thank you, sir.
Thank you so much, Dr. Chaudhary and Dr. Chitis. Bye. See you.
We shall meet tomorrow.
Yes, sir. Thank you.
Bye to
Audience, we
all.
can meet tomorrow
Good
morning.
evening, good night. See you
Yes.
tomorrow.
Okay, sir.
Can I close, sir?
Yes, you may close.